Hello, 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 hello. ज्वाइन करेंगे साउंड बढ़ा दीजिए.
Okay, so let us invite Dr. Nitin Kulkarni, Director, Institute of Forest Productivity, Ranchi, for his talk. Dr. Kulkarni, are you connected?
Yes, sir, I am connected. Am I audible?
Yes, very nice. We can see you.
Okay.
We can see you there. Kindly, you can begin with your presentation.
Okay, okay.
Want to cast your screen, you can.
Sure.
ठीक है सर.
Is it visible?
Yes, now can you make it full screen if it is possible?
Pardon?
Can you make it full screen? Please?
I have already made it full screen here. It may take a few seconds there, I think. Let me know when it is ready. Is it visible now?
We can see your screen and we can
ask the share entire screen.
Whether the slide is proceeding, please.
slide is not moving. We can see only the first slide. Insect diversity, economics, and conservation. I think you please disconnect and again share your screen.
Okay.
Next slide हो गया.
You can share entire screen instead of a window actually.
Okay.
That might help the problem. Yes.
Is it visible now?
Yes. Can
And it is moving on.
Yes, yeah, very nice. Yes.
Okay,
That-
okay.
all set. Please go to the slide. Thank you very much. You're welcome to present.
Okay. First of all, today's chairperson of this session, Professor S.P. Singh Saab, co-chairperson Dr. Virtually, so I can't see all the faces physically present there, but I must thank first of all the Guru Ghasidas University Administration and the Professor Singh also for giving me this opportunity to discuss this important topic, that is insect diversity, particularly the economics and the conservation. I am in fact delighted to present this topic because this particular aspect, that is insect diversity, is nowadays particularly is very much required to be discussed and given due emphasis. With this, as I was told earlier that only half an hour is given, accordingly I have tried to make the topic justifiable so that I can discuss more things in the given time. However, the diversity itself is such a large topic that it requires time. Anyways, I hope I'll be able to justify it. Initially, I'll discuss, I have divided this presentation into several parts, like as the title also says, some of the hardcore things about the insect diversity, global, national, and as well as the Central India also, because Central India is the theme of the this conference. And then the diversity from the economics aspects also, and as well as why we should go for conservation aspects. That is also briefly discussed here. So with this, I'll start the discussion. Now, I don't know that whether the entomological aspects are being is a part of GGU curriculum, and if it is there, how much it is there. But even then, it is interesting to know that when we discuss the insect diversity, we must be aware that insects are present everywhere. We cannot imagine that the insects are there in the glaciers also. The insects are there in the volcanoes also. So these are the climax ecosystems, you can say, which are actually disturbed because of the natural phenomena. But there also we find the insects. You can see some of the examples where the lady beetles are also present in the volcanoes. And similarly, the carnivorous caterpillars from the volcanoes in Hawaii. So this is the reports from various parts of the world where the such events have taken place, and in such difficult areas also the insects have shown their presence. Like the tent caterpillars have been noticeable presence, particularly in the crater and around the rim of the volcanoes. So with this, we can say that when these extreme places can be inhabited by the insects, then why not others? Now coming to the world scenario, when we say the insect diversity, then definitely we must know that on an average 55 lakhs, this is an average based on the statistical calculation, and the range is from 26 lakh to 78 lakhs of insects. That is 75% of the global fauna are available in the globe. Although 68 lakhs are the terrestrial arthropods. So this is actually, these are the figures which have been calculated based on statistical calculations, and these are the recent figures of 2018 by Stark, which by using various methods, particularly the statistical calculations, have come to the conclusion that there would be 55 lakhs of insect species in the world. Now, what is interesting is that only 10 lakhs, almost 20% of the insect species have been named and they have been reported. and some of them have been described also. The least studied taxa are the Coleoptera, Diptera, and Hanoptera, and therefore these are to be focused more for remaining 80% of the expected populations. So this is actually a very interesting data which says that even if we have already got the 10 lakhs of insects already identified, they form only 20% of the expected population. I'm using the term intentionally, the expected population, because 55 lakhs is expected population based on the statistical calculation using the parameters like the availability of the host species and the insect pests reported on hosts. So like these other parameters have also been considered for that. So 80% of the expected populations are yet to be studied, reported, and described. Now, it is very interesting to know that 1 to 2% of these populations may be truly cryptic. Cryptic means their habitat is so hidden and cryptic that maybe their identification may take time also, and it depends on the researcher in the various parts of the world as and when they report these things. With this, if we see that the 29 insect orders, if we see starting from Mecoptera to the Archaeognatha, we find that this is the distribution of the species which are world over known. The figure is already there. I'm not going to read these figures, but the total is something around 10,13,825. Now, it is very interesting to note that every time we visit the literature, we find that 10 to 15 insect species increase. And that is because the researches are continuously going on. Like in the slides to the proceeding slides, the figure will slightly change because the reference which I have used is recent one. And this is from 2018. But this is the tentative figure, because whenever we say worldwide, that means the figure to collect at one place is really very difficult. But our people have already done it, and this is the figure which can be said that the availability in the which have already been reported. This is the 20% of the expected insect populations in the world. Now we also know that the India, the mega diversity hotspot, we say, it has unique biographical locations. It has diversified climatic conditions. We all are aware. It has varied ecosystems. And that is actually, these are the reason why we have the mega biodiversity hotspot status. And the two of the major biographic regions are Palearctic regions, that means somewhere on the foothills of Himalayas, where the pine forests and others are there, and the Indo-Malayan. Indo-Malayan region means the most of the country is covered under this major biographic regions. Then we can say that three biomes are there, that is tropical humid forests, tropical dry deciduous forests, warm and semi-deserts. And the map shown here is actually, there are many other types are also shown, the forest type groups rather. So these are the further classifications of the forest types. Now audience must be slightly surprised that why I am discussing about the forests rather than the insect diversity. That we'll come to know in the proceeding slides. Now if we see this particular slide where the Indian fauna from the world has been shown. From the data which has been shown earlier also, considering that, we can say that the total Indian fauna is 1,02,161, which is 10% of the total world fauna, particularly insects. Out of them, the arthropods form the 76% 1,461, that is something around 74.8%. And the insects, purely insects, because arthropods, we are aware that there are many other groups also which are arthropods. But purely if we see the insecta, then it is 66,911, that is 65.4%. And this data is based on the Chandra 2020 as per the reports of Zoological Survey of India. So this is, as far as the available data is concerned, is the updated data on the entomofauna in the country. Now, if we again distribute this available data in the different orders, available orders, then we find that based on this 2020 data, we find that the entomofauna recorded for different orders, insect orders, is if it is presented in a graphical format, then it is like this. The highest population which we have is the Coleoptera. Then you can see the right side of the figure, which is actually about the Indian reports. And as per this, the highest is Coleoptera, which is with 22,334 species known, followed by the Leptoptera, which is 13,694. And then the Hymenoptera, which is 10,605. And then Diptera and Hemiptera, etc. And this trend is in line with the global trend. That means the populations in these species is very high, because if we see the global population also, the known population of Coleoptera in the world is 3,86,755. This is amazingly high, diverse conditions as far as the insect order is concerned. And that is very true also because of the Coleopterans being very rather highly adapted insect order, which can survive in various given ecosystems. Now, if we must be actually interested to see that what is the faunal status of insects in different ecosystems. If we tabulate this in the form of the table which has been shown here, then we find that agro ecosystem, the insects is 3,130. Freshwater is more, it is 4,976. But the highest is in forests, and that is why even if we do not want to talk about forests, say for an example, even then we have to talk about the forest, because the highest number of species which are known are from forests only. Due to the obvious reasons we all know. So it is around something 63,733. And if we compare it with the percentage against the insect fauna of India, then it is 95%. 95, almost 95, more than 95% of the known insect population is from forests. Whereas we mostly consider the agro ecosystems and other ecosystems as the most inhabiting ecosystems, but it is actually not. Forests have the maximum entomofauna available as per the records. So that is why we have been discussing forests, and we cannot leave forests, because this is the place where we have to give most of our attention. Having discussed the diversity aspects, these are the data which are available. Otherwise, if you see the enormous number of the population of insects world over and countrywide also, it is not possible to show all the insect pests which have been reported here, and even to list out the insect names also in a given time. Therefore, the data which has been shown itself is sufficient to show that how good diversity we have, because we have 10% of the total populations available in the world. And probably that is why we are the hotspot. Now, one may ask the question that only by insect populations, how can we be so rich in the diversity? But I must add that it is not only by the number of populations, but also by the economic services of the population, which actually determines that how important they are. Secondly, insects are actually the, if you see the food chain and food web, they are the primary consumers, and therefore they form the base of the basal part of the food web rather, and therefore their presence actually determines the health of the given ecosystem. So these things are very important, and therefore I have listed here the to discuss the economic services also and the environment services. That is the environmental services by the insects. I have divided them into the harmful insects as well as beneficial insects. Now, harmful are those which normally we see, that is the insects as pests, medical and veterinary insects, invasive alien species. There could be hundreds of more, but mostly, majorly, we can divide them in these three major categories. Coming to beneficial insects, if we see, then definitely the pollinators, which are actually the most important economic services, economic and environmental services rather, delivered by the insects to us. The food security, insects as natural enemies of pests, because those who are not from entomology, they understand that insect pests are the harmful insects which feed on the plants, that is herbivorous insects. And the insect pests as natural enemies means the natural enemies of insects, rather natural enemies of insect pests. So that is why they become beneficial to us, because they are feeding on the harmful insects. Then insects as nutrient cyclers and insects as environmental indicators. I'll discuss a few examples here as whatever time which is available to me. I'll go through some of the presentations, some of the examples rather, one by one. Now we first of all see some of the important examples of insects as pests. Now, the central India, we all know that the MP, Chhattisgarh, and many others, agricultural pests we all know very well, because there are rice pests, there are many other pests. But let us discuss something which is normally many people may not be knowing, because Chhattisgarh is also the state where the sal seed trade has been very common, particularly until before the Supreme Court order after the sal borer outbreak rather. But I think it is still going on. And this particular seeds are actually infested by many borers, and they cause economic losses to the precious seeds which are actually collected by the tribal and given to the traders for soap making and many other useful industrial importance. The upper right, leftmost, you can see the healthy seeds, and right one is the damaged seeds. Below left, you can see the insect which is damaging it and completely making it hollow. And the right lower picture shows that how eggs are laid by curculinid, that is Cytophilus rugicolis, inside the seeds, even while they are on the trees. And that is how the seeds are damaged completely. Another example of harmful insects is the large nurseries. You can see the teak, because we are actually in the teak belt. Presently, I am in Ranchi, that is eastern part, but in central India, teak is the major crop, forestry crop rather, and which is grown in large nurseries, maybe extending up to 30 hectares and something like that. You can see the pictures, the enormity of the forest nurseries, and all these nurseries have a common problem of white grubs, which actually damage the teak seedlings. The leftmost picture, you can see how the wilting is observed, and an insect is shown, which is actually the adult of the grub, that is the Holotrichia rustica, and how this immature stages feed on the roots is also shown, and ultimately how they damage the seedlings is also shown. So this is how these are the major insect pests. I have included only the major insect pests here, which are economically important, leaving the minor insect pests which are damaging or simply defoliators like that. Just to emphasize the economic importance of the insect pests also. Now here you can see the sal borer on Shoria robusta. This is actually not a borer affected area, but the whole Chhattisgarh, MP, and even Jharkhand also, where presently I am stationed. This is actually a major sal forest area, and you can see a healthy forest how it looks. But if you see, once the infestation is there, you can see the picture that this is the picture taken by me in Chilpi range during 2014-15, when the sal borer infestation was at the peak during recent outbreak. And you can see in insect, the female and male insect adults of the sal borer, that is Hoploceramix spinicornis. Now these insects are causing heavy damage to the trees even while standing, as well as in timber depots. The below picture is self-explanatory that how multiple tunnels are made in the tree bole, and the whole bole, even if it is above 100 or 200 centimeter, even then it is of no use. But other than the of no use, then rather I think it can be used only as fuel wood. So the value is completely gone, and this is how the adults are also collected. This is actually a picture taken at Tokapal, Jagdalpur, where our team visited for the observations and some of the remedial measures. Now, this actually is the major culprit, because particularly in case of sal borer, the grubs are the major damaging stages. They cause tunnel inside the tree bole, and that is how, because of the multiple tunnels, the trees start drying from the top. Here I am not explaining the complete life cycle of the insect because of the kind of topic which we are presently dealing with, but simply would like to show this important insect at length, because this is the most important insect pest in the central India, wherever the sal is there. This is the female and the male adults of the sal borer. Now, as per records, during the 1998-2001 outbreak, nearly 70 lakhs trees were required to be removed from the forest areas. As you can see this picture, when we visit the forest areas for the training purposes of the frontline forest staff, there this picture has been taken. You can see that the wood dust gathered around the tree bole, and this tree is actually completely dead, although the bark is intact but completely dead. And these trees have to be removed from the forest areas to prevent further spread of the insect pests, and this particular sal borer rather. Now, coming to another important example in the central India, this is about the teak defoliators. You can see the kind of damage caused by teak defoliator in the teak plantations. And this is the where the complete leaf defoliation is visible, and this hampers the overall growth of the teak trees. As per the Beeson 1946, I remember the cumulatively 5 years, if this kind of insect pest attacks are there, then they may affect from 13 to 46% of the increment growth in the teak. Various experiments are going on as to develop the chronal materials which are resistant to these insect pests, but still the work is in infancy, rather, I will say, because of the complexity of the clonal development in teak. I think many of our participants, those who are working in forest genetics, they understand this problem. This is another insect that is teak skeletonizer, that is Eutectonomacrylis, and you can see a beautiful picture. Although it looks good, but this is very damaging because cumulatively teak skeletonizer as well as the defoliators, cumulatively they cause increment loss in the teak stands. This is the adult, and this is the lower one are the larvae of teak skeletonizer. Now, if you, as we have listed there, here I'll be showing some of the natural enemies of the harmful insects. The nature has given everything. Unfortunately, the natural balance we have, the human beings have rather disturbed it by way of various our activities in the forest and around the forest and in our surroundings. This is actually a saw borer predator, Allos sordidus, which used to control the saw borer earlier, in old days probably, because the population of these predators as well as the saw borer was in balanced situation, and during the outbreaks period, it is found that the population of these saw borer predator, that is Allos sordidus, is reduced, and that is why probably the outbreaks occur. Although this is not the only factor, there are other environmental factors also which determine the outbreaks. I have also shown here the white grub stages in the forest nurseries where the white grubs damage the teak seedlings, and this is actually the perfect example of Agrippinus, the predatory example of white grubs, that is Agrippinus fussipus. One can see here the white grub which is half fed by this larva, the elaterate larva of Agrippinus fussipus, and this I have recorded from Nagpur during 2017. After 1947. So this is the first report after 1947 where I have tried to study the whole biology of this insect pest, and this has been extended to the forest nursery staffs that please do not kill this insect because they are unable to differentiate the larvae of this elaterate beetle as well as the white grub beetles, and they unknowingly they have been killing this beetle also, and that is why the population reduces and the outbreak of insect pest also has been recorded. So this is how these kind of things are also very helpful. Similarly, in Tropical Forest Research Institute, where I was working earlier, we have developed the tricho card. These are the perfect examples of, rather this is actually a perfect example of egg parasitides, because they are parasitizing the eggs of the insect pests. So right side you can see the top picture shows that how the eggs produced inside the laboratories are pasted in the cards, and these cards are available, these are on sale, available from Tropical Forest Research Institute. These can be transported like the picture shown here in the left lower picture, where the cards have been placed inside the polythene packets. They are stored in the fridge, and whenever they are to be released in the forest area, they are simply taken and the cards are taken out and released in the forests. These egg thrustoids, within 2 to 3 days, they emerge from the eggs and they parasitize the harmful insect pests of teak defoliator and skeletonizer. So this is actually a perfect example how we have already made use of the natural properties of egg parasitoids. That is, in this particular case, it is Trichogramma raoi. There are other exotic species also available like Trichogramma chilonis, Trichogramma pretiossum, and many others. But this has been recorded from the Central Indian forest floor, and therefore this species is very important, which we have already made use of, and this is being supplied to State Forest Department of Madhya Pradesh, and probably Chhattisgarh also is getting training on this by the Tropical Forest Research Institute. And this was the work which we had carried out during our tenure in TFRI Jabalpur. Now this, you can see the result also after once the tricho card is released. The left side is the damage impact where the cards were not released, and right side you can see the release side, at least 50% damage has been prevented. And whenever we say the biological control, that means the even 50% management is also a good result, we can say. Similarly, there are other examples also like Canthicona fuxillata, which are actually the nymphal stages as well as the adults. Both are predators of insect pests, and they are also being utilized now commercially as predatory insects. And in TFRI also, I remember we have already started the mass multiplication of these insect pests. These are other examples of predators. I will not go into the details of this, but the picture themselves are self-explanatory. This is the white grub, and this is the predator insect, and how you can see that it predates on the white grubs in the laboratory. Although these are some of the minor predators, but even spiders cannot be neglected, because these spiders are available in the forests of Central India, and they play their role in managing the insect pests. As we are not actually only discussing the insect pests and their biological control, but we are actually discussing the biodiversity aspects, particularly from the economics point of view and social services, and economic environmental services point of view. Therefore, this insect is also very important to be discussed here. This is Zygogramma species, which is actually a biological control agent of Parthenium weeds. You all know the harmful effects of Parthenium. This photo I have taken from the Motinala range of Madhya Pradesh and Chhattisgarh border, just near the Chilki, where this was naturally found. Otherwise, this insect was earlier imported from abroad, and it was introduced in a given area. But I was amazed to see naturally getting established in the forest. And this is actually something I can say a good news, because maybe in future we will be able to make these insects also use as biological control of Parthenium and other weeds. Now coming to the other economic aspects, these are the textiles from insects. I've given the title, but actually it is this particular photograph shows the Antheria mylitta adults and its larva, which feeds on the asan, that is Terminalia tomentosa and Terminalia arjuna also, as well as the sal also. And these are actually the most important economic activity, particularly in our Chhattisgarh as well as the Jharkhand. Here also, you all must be knowing that the Jharkhand is also the state which is leading in tussar, similar to the silk from the silk moth in various parts of the country, particularly in south. But in this particular part, the Vanya silk is very dominantly and prominently practiced as socio-economic activity. among the villagers. The villagers, they collect the cocoons from the forest and sell it. We must not forget the diverse honeybee populations from the surroundings. We have a long history of honey consumption. As far as our present studies go, presently we are consuming only the honey from the Italian honeybee, that is Apis mellifera. But actually, earlier we used to have the wild honey, that is honey from Apis dorsata. In this particular picture, you can see the Apis dorsata honeycomb in the natural forest, and this can be actually, although it looks very dangerous, sometimes it is dangerous if disturbed, but otherwise, this is actually a sort of, you can say, an amrit with medicinal property for the human beings. And we all may not be knowing, but the India is known for wild honey, and the wild honey is from the honey from Apis dorsata, that is this particular picture which I am showing here. Otherwise, the cultivated honey is from Apis mellifera, which is also good, but it comes next to the wild honey. Coming to the lac insect and lac culture, we are next to China, of course, but second in the world in production of lac and lac cultivation. Again, the Jharkhand, particularly in this part, is very important. In central India also, we have been working on the lac cultivations. But if we see worldwide, then India stands second, and if we see countrywide, then Jharkhand stands first in the production of lac. Of course, there is competition between Jharkhand and Chhattisgarh for the production of lac, because both are the adjoining states, and sometimes the cultivation is more in Chhattisgarh also. But presently, again, I think the Jharkhand is leading. So this is the most important economic activity of the insects, which take place and gives employment to and livelihood to many of the tribal in these areas, because these can be reared in Paras, Bear, and Kusum for various kinds of strains which are available. Now, these are some of the uses of lacs which I had just included, but I will skip for this time. Now, the insects as pollinators. This is one of the most important environmental services which insects play for us. And we all are aware that although the honeybees are the most studied insect populations because of its various services, including economic and environmental services. But the pollination aspect has not been that much given due emphasis, particularly from the multiplication of honeybees and introductory releases. Because my studies earlier have shown that the various situations of in MP and Chhattisgarh in various parts, thousands and thousands hectares of trees are of one species only. Like teak, if it is planted for thousands of hectares. So this is actually a monoculture, and it disturbs the natural balance of year-long food availability and nectar availability for honeybees. This is actually reducing the population of honeybees and diversity of honeybees in all the areas. It is not only the case for the central India, but for the whole country and the globe. Other major factors are the use of insecticides, agrochemicals, which are actually disturbing, because these agrochemicals and chemicals also reduce the population of these honeybees because of the negative effect, and that is why they are heading to mass extinction. Some of the species are now going to be extinct, and this is very dangerous, and therefore we need to conserve these species. And that is what has been given here. I'm not going to read out, but definitely more studies are required to be done, and that is why in central India we carried out some experimentation in the forest area where the population of honeybee was less, and we found that while the tea stands, their seeding percentage was getting lesser and lesser every year. This is actually the area in Behrai Peak Seed Orchard in central India, near Jabalpur. And when we took the observation, we found that the overall data every year was reducing, and most probably the reason was the reducing population of the honeybees there, which actually help in utilization. And therefore, we carried out an experiment here. We observed the fruiting percentage earlier, and after the honeybee release in the area. So we rather introduced the honeybees, that is Apis mellifera, in boxes in the forest area, and we found good results, considerably good results, and good seed setting in the area where we released the honeybees. So this is actually why it is important, because honeybees actually are not only the economically important insects only for honey, but they are actually the insects which are actually also doing the environmental services, most important environmental services, by increasing the productivity of forests and the given crops. Not only forests, but even the agricultural crops also, and that is why they must be conserved and protected. The next in economic activity is insects as food. Now, FAO has already said that the insects are going to be the next alternative for food. That is why it is they have given the title, "The Human Bites Back," because earlier the insects were known to bite the human being, but now humans are biting back. So entomophagy, this is also called the entomophagy, which is very interesting to know. But although many people do not like it, may not like it in today's time, but maybe in future we will be compelled to do that if we do not understand the importance of the problems which human beings are themselves creating for their next generation to come. So this is how the entomophagy has been there, with 1,600 species of insects already consumed by over 300 ethnic groups worldwide. And in general, they are species-wise, if you see, 235 butterflies and moths, and 344 beetles, 313 ants, bees and wasps, as well as other species also are known to feeding considerable population in the world. I remember in Chhattisgarh, because I was in Akir long ago, it is a practice to collect the red ants from the mango trees, and then people used to make chutney of that ants, and this is how this has been in practice since beginning also.
If you are hearing me, can you hear me, please?
Hello,
या आप मुझे सुन पा रहे हैं?
हाँ जी सुन पा रहे हैं.
हाँ, आपसे थोड़ा रिक्वेस्ट है, अगर थोड़ा सम अप कर दें थोड़ा देर.
अच्छा, ठीक है. Okay, then this is the entomophagy, which is in the recent time, which is the examples which I have shown, so that people know that what kind of edible forest insects are available in the world. And I'll, as the chairman sir has already given me a notice to wind it up, I'll go faster. These are the number of edible insects per continent which are known, of different orders. These figures are very interesting, but it is available. One can see the purpose is only to show that the insects have these economic use also, because as food also they are going to be future. This is the picture from the Southeast Asian countries where the insects are very common. You can see the colorful street market where the insects are being fried like samosas, and they are being supplied to rather for them it is very delicious. This is the example of weaver ants which I was discussing, that it is available in Chhattisgarh. Now there are some insects which are very useful as aesthetics and environmental indicators. Now I'll not explain the bio indication required because we all know that the environmental situation is getting deteriorated day by day, and we need to understand the environmental conditions by way of only seeing the populations there, and that is why these are called the bio indicators or environmental indicators, and particularly the Lepidoptera, that is butterfly and moths, are the best bio indicators, and therefore these should be more and more studies should be taken for that particular given ecosystem, that which insect species is actually the bio indicators. Now, some of the important species insect species in the central India, I'm just showing the pictures. I'll not go into the details, but this is how the diverse populations is available in the central India, and this particular many of these species are from Kanha National Parks and various other areas in the central India. Now, these are some of the pictures which are beautiful examples. Just I have shown to for the information to others. These are not actually the Indian populations, but very beautiful, aesthetically important insect species. Similarly, the insects also play a role in nutrient cycling. We all know the termites and how they. We only know the harmful effects of the termites, but we must also understand the ecological services they play to the environment by cycling the nutrients available in the environment, and this is how they're also useful to us. We should not look at them only from the pest angle, but also from the nutrient cycling angle also. Now, what are the threats of changing conditions? We have only 2 slides, I think. I'll be finishing in 2 minutes, Chairman sir. So these are actually habitat loss, deforestation and degradation, agriculture, encroachment of forest land, grazing, human wildlife conflicts, replacement of indigenous species with exotics, imbalance, infrastructural developments. And then IUCN Red List we already have, which is updated up to August 2020, which includes 22 species from India, which is already under the on the verge of extinction. This is very alarming because as on August 2020, we find that in India 560 species we have already lost. So that is why this actually gives an alarming tale rather for all of us researchers understand the importance of the diversity in general, biodiversity in general, diversity of the insects in particular, and the role of environment in forests and various other ecosystems where the healthy status is to be maintained and which is very much required. Although the efforts from the government angles are going on, but every individual's responsibility is to support this kind of activities where we maintain the natural balance between the good and bad organisms and the environmental conditions, so that the diversity flourishes in the future, not only for the insects but for the other animals also, other organisms also, and only then the human beings will be surviving. And that is why I have included this slide which shows the backdrop, in the backdrop, a good forest area, which is actually a sacred grove from Jharkhand, which shows that how healthy forests still can be managed by the local populations. This should be promoted. And thank you very much. And we must remember that we have only one earth. Thank you. Thank you all.
Thank you. Thank you, Dr. Nitin Kulkarni. Wonderful presentation and very illuminating from the academic point of view as well. I thoroughly enjoyed. Let me introduce myself to you. I'm a Washington from Nehu Shillong. So I am a professor of botany there. But I see a lot of insects around me in my house, and then just take some pictures of clients out of curiosity. Of course, I am not an expert, but we have plenty of diversity there. Some of the insects pictures you have shown are related pictures are there in our places. There is a wonderful diversity of insects. As we all know, insects is one of the largest groups of organisms and the least explored one. You have mentioned already, not more than 20% of insects present are known to the science. And we have a lot of opportunity to explore more newer number of species. And this is a message to the youngsters actually. This is one area in which you can make a name by exploring more and more. And also you mentioned cryptic species actually, which is very less known to the students, and there is a lot of cryptic species diversity as well, which is found. Insects can be beneficial, insects can be harmful. These aspects you have illustrated very well in your talk, and how sal borer has been different groups of insects are damaging sal forests, sal timber, sal seeds, etc. This is very big problem. So how to find enemy for enemy is another area of research, I think, so that we can tackle these problems. Of course, there is a lot of scope to work for protecting our timber yields and sal seeds. Yeah, I think we have learned a lot from you, and we will benefit more from you in future. I will be interacting with you, Dr. Kulkarni. Thank you very much for taking time out for this audience, both online and offline. Let me ask if there is anybody who has any curiosity to ask you a good question. Anyone, please? Yes, there. Let me say online. Is there any online person who would like to ask? Okay, if not, then there is one offline.
Hello.
Oh, yes, please.
Myself, Dr. Hariom Saxena, from Tropical Forest Research Institute, Jabalpur. I would like to thanks and congratulate Dr. Nitin Kulkarni sir, who is my senior, who was earlier in TFRI. And I thank you, sir, for a very wonderful presentation, sir. Thank you, sir. Thank you so much. I extend my best wishes to you, sir.
Thank you, Dr. Saxena.
Sir,
Okay, so
thank you.
here is some
Thank you.
Good afternoon, sir. I am Ashutosh Anand, research scholar from Department of Forestry, University University. Sir, actually, I have a question regarding the natural control of pest. As you were presenting that there are some insects which act as a natural pesticides. So, when we look in the history, there is an incidence that a rapid increase of population in the insect pest harming the crop and plants. So there is also a natural predator, praying mantis, which is polyphagous in nature and has a wide range of prey. So is it possible to artificially raise that praying mantis in control condition, and when such insect outbreak happen, so we could release them to those areas and to just control the such outbreak? Is it possible to artificially raise those praying mantis?
Yes, sir. May I know your name, please? Your voice has been breaking inside.
Ashutosh Anand.
Acha Anandji, actually, your question is really very good and very important also, because this is the actually line where we all must consider our future actions in the future to come, and it is very much possible. Only thing is that you have to first expertise yourself in maintaining the laboratory populations of the mantids, and then see that whether the introductory things can really come up. Now, I must actually just give you some tips that as we say that it is possible, it is definitely possible. One has to experiment. That is why it is research. But it is not going to be very easy also. There are various aspects which one has to understand. And that is, first you have to develop the mass multiplication technique of the mantids in laboratory. And then you have to see that how that can be transported to the field. Because, and in what conditions, whether in the egg condition or nymphal condition or adult conditions. And then only you can, and then ultimately, what is the biological control, practical biological control in percentage terms it gives you in the field against the particular insect pest. Once it is studied, then only you can calculate the economics of the mass multiplication techniques and the introductory release. But yes, it is very much, unless we think on these lines, we will not be able to even experiment. So I just hope that you will be able to do that in due time, and definitely I am here to discuss all these things whenever and as and when you need.
Thank you.
Thank you.
Exactly, and Dr. Nitin will be very pleased to.
Sure, sure.
Thank you. Thank
Yes,
you,
sir.
Thank you very much.
Thank you, sir. Thank you, Chairman, sir. Thank you.
Thank you once again. So we invite the next speakers now. Okay. So let's make next now. Please, organizers, please tell who is going to be called. Dr. Hariom Saxena.
Yes, sir.
Yes. Are you able to present now? Are you
Yes, sir.
ready?
Yes, sir. I am actually I am sitting in the car. Actually, I am on the way, so that's why I am
All
going.
right. All right.
So,
All right.
sir,
We will
sir.
be pleased to hear you. So please
Okay,
begin.
sir. Sir, am I this presentation is visible, sir?
Yes, your screen is visible wonderfully well. Please
Okay,
go
sir.
ahead.
And my voice is also clear, sir.
Yes, yes, yes.
Okay. So, thank you,
Moreover,
sir.
I will request you to give the salient points of your talk and,
Okay, sir. Okay.
and conclude, I think, focus more on the objectives and in the theme of the work
Okay.
rather than the nitty-gritties. So
Okay.
it's,
Give me only 5-6 minutes, sir. 5-6 minutes. I will conclude, sir.
thank you so much.
Okay, thank you, sir. First of all, I would like to thank the organizers for giving me this opportunity, and I would like to thank the chair and co-chair, sir, that was accepting my presentation, because I am in the field, so it was not possible for me to get connected with the internet, sir. So thank you, everyone. My work is the chemical and morphological variations in stereosome surveillance. A vulnerable species, related species in Madhya Pradesh, and its issue conservation, sir. So this is the species Utilisation surveillance, sir. Height is about 20 to 25 meter, and commonly known as the trade name Padar Patala, Ada Sisi in local languages, and the commercial part which is used in the pharmaceutical formulations is the roots, and which is used in the formulation of the small rest. And you can see here that the small formulation is not only used in the formulation of the solarist, but the basic formulation, basic ingredients of the 10 species is also used in more than 100 of the Ayurvedic formulations. And some of the formulations are given here also. And this is distributed, the plant is distributed throughout the sub-Himalayan tract and outer hills, Rajasthan, Chota Nagpur, Central India, and Western Peninsula, and Bangladesh, and English forest also. And this is one of the priority species which has been identified by the Ministry of Health and Family Welfare, Government of India. And government is also providing the 50% subsidy for its cultivation under the scheme of National Ayush Mission. This is one of the species of the smaller dish, so the actually root bark of this species is utilized in the herbal formulation. The rate of this the dry bark root is dry bark of the root is 300 rupees per kg. And each and every part of the species is very important. Bark, pods, flowers, leaves, everything is important and used by the traditional healers and have pharmaceutical potential. And important for the used as a anti-cancer, anti-diabetic, and in the treatment of leprosy also, sir. And you can see here, this is the some of the flowers and seeds and parts of this tree, different plant parts. And why I have taken this history? Because this species has become rare in Madhya Pradesh, and also notified by the Madhya Pradesh Forest Department. and it's a very fragmented population of the species is reported in Madhya Pradesh, and this is rapidly declining from the natural pockets. And destructive harvesting coupled with reduced seed production also one of the reason for the fast erosion of its natural population from the forest areas. So, as we know that if we devise any conservation strategy for any vulnerable or rare species, we have to know the distribution, natural regeneration, chemical and morphological variation, what type of diversity is there. If the plant is important, it have the pharmaceutical importance, then the chemical evaluation and evolution of the chemical ingredients becomes the more important for screening of the better chemotypes. So we have did the chemical profiling also, and we have did the morphological recorded the morphological characters also. So this you can see here that why this species is distributing from the forest, because you can see here that the people are not knowing well about the species and they are cutting it. They are for various purposes, for fuel wood, for means for making some furniture in the for their home. So this species is one of the region they have recorded this thing in a picture from the Khandwa region. So the methodology I have adopted that survey we had survey from forest area assessment for the variability of important morphological traits we recorded. Then we collected the root samples, stem leaf. We also quantified the chemical content in the stem and root and found that if the chemical content are found in the stem and leaf, then can we use the root and stem, the stem and leaf in place of the roots. That is also alternative plant parts we study the alternative plant parts to explore the possibility, and quantification of active chemical ingredients and establishment of germplasm bank to conservation. So we have demarcated 102 trees from the different 9 forest divisions of Madhya Pradesh, and GPS readings were recorded. And this is using the high performance singular chromatography. We have quantified the tricontinol. Tricontinol is one of the major phytochemical ingredient and active ingredient found in this root of this bark plant, and it is also mentioned. This is the phytochemical marker as per the quality standards of Indian medicinal plants published by the ICMR. So you can see here this is the 3D diagram, and all the tracks the tricontinol present. So in samples as well as the standards. So this is the methodology SPTLC was used for the quantification of tricontinol. And you can see here that quantification of tricontinol in root bark samples of SPTLC, the Khandwa region, that region is found to have the highest maximum content of the tricontinol, 0.54%, and the lowest was found in the Mandala region, 0.12%. So this is the table shows the recorded morphological traits and the tricontinol percent in the external surveillance collected from the different regions: Mandla, Hoshangabad, Siuni, Jabalpur, Damoh, Khargone, Khandwa, Balaghat, and Chhindwara region. You can see here that we have also correlated the intercharacter correlation between the different traits of the surveillance species. And you can see here that the important characters, we try to correlate the important morphological characters with the tricontinol content. So the correlation analysis revealed that the tricontinol content showed positive and significant correlation with the tree height, girth at best height, single bole height, and number of flowery branches. So if we are going to select the CPTs or to select the morphological superior trees, then we have to see that good means a good number of that tree height, tree girth, clear bole height, and number of primary branches should be good. Having a good number of primary branches should be there. So this is and after conducting this study, we have established the germ plant bank in our institute, having near about 1010 plants, 110 plants from the 10 locations of the Madhya Pradesh, and we established in 2020 itself. So and this germ plant bank is having all the diversity. Wherever we found the plant, we collected the seeds, and we also and we just conserve it in the germ plant bank here. And so this city provided the chemically and morphologically characterized germplasm of this species, and the superior trees can be conserved for the feed source. We have also informed to the forest department that where is the superior trees and where they need to conserve this tree for the further analysis as well as further study, as well as for the collection of the seeds for the preparation of the seedlings in the forest extension nurseries. and the information generated through the species will be of utmost importance and will serve as a baseline data for devising the conservation as well as the future management strategy of the species. And the established germplasm bank of the species will help in its conservation and further studies, sir. So this is all about my small studies, sir. Thank you very much for giving me the time. And if you have any question, you're welcome, sir. Thank you so much.
This stereoespermum swabiolens is where it stands in IUCN category. You have mentioned RE3 species, right?
Sir, it is regionally RET because the forest department Madhya Pradesh have notified this species as a vulnerable species. They have so I have said it is the regionally means at major regional level it is the RET.
Yeah, but IUCN status also you can mention in your study.
So, in IUCN status it is not given, sir.
Not great.
Not included, sir. Still.
No, it should be there. Some maybe either vulnerable or diet deficient or something.
Sir, I have, sir, I actually set out, but it is not there still. Means maybe it will not be there, sir.
Anybody online would like to interact with Dr. Hariom Saxena? No, I think so. Thank you very much for your very nice presentation on the species which is of paramount importance in Central India and deciduous forest. So thank you, Dr. Saxena.
Thank you so much, sir. Thank you, sir. Thank you, sir, so much. Thank you. Thank you.
So next speaker,
नेक्स्ट देखो उसके बाद फिर मार सकता होगा एक अभी है यहाँ पे और क्या जो भी जिसमें
Dr. Vivek is connected. Yeah, Mr. Vivek.
Yeah, I'm available, sir.
Yes, Dr. Vivek Vaishnav.
Yes, sir.
Yes. So it's your time now to tell us and share your experience.
Yeah, I'm facing some kind of echo. The sound is not okay. Hello, am I audible?
Yes, please.
Yeah. Thank you so much.
Can you please make it
Respected.
if you can
Yeah.
larger font size so that everybody. Yes, wonderful now.
Yeah, thank you so much, respected chairperson, the dean of the school, the head of the department, respected professors, teachers, dear scholars, and all the participants. A very good afternoon, and thank you so much for giving me this opportunity. This work that I'm going to present here is based on the tree improvement program, and specifically the genomic application on the genetic improvement of the teak, initiated by the genetic division of Tropical Forest Research Institute. So everybody, it is now generalized that teak is a major species of central forest in India, and most of the times when the plantation program is being conducted through the Van Mahotsav or any kind of afforestation program, teak is a major species that is planted. Apart from that, the genetic improvement program initiated in India in 1970 with the teak. So Paragon, it is known because of its timber quality. Everybody knows naturally it occurs. It is called as an endemic to South Asian countries, India, Laos, Myanmar, and Thailand, and later it is introduced in other countries as well of America, Europe, Africa, and Australia. So one molecular marker or DNA marker based study was also conducted, and it was found that genetic origin of the teak may be from the Indian subcontinent, specifically South India. This study was carried out sampling the teak from all endemic, all the countries where teak is having endemism, means Laos, Thailand, and North India. So basically, if you see in Indian perspective, India represents 43% of the global teak genetic resource. But if we'll see the maximum mean annual increment, if we talk about the productivity, it is there in Indonesia, not in India. India is producing only 12% increment every year. And we are producing 49,507,000 per meter cube quantity every year, and list of that quantity we are importing, and still we are 6 times of our import is means it is coming from the Indonesia country. Okay, so there is a very huge variation in the export and import. Only 43% we represent, means India represent the genetic resource of teak, and we are not able to export the justifying quantity because of heterogeneous genetic resource we are having, and we need a rapid selection. And because of that entire breeding program, if you see that is based on the selection and then crossing and breeding, it may take some high time. That's why molecular marker technique is a kind of solution that if you are introducing it for the selection can be done at early age, and we can reduce the time for the genetic improvement of teak. So with this background information, we decided 4 objectives. First, evolution of the existing genetic resource variability, gene pool characterization of Indian teak meta population. Apart from that, how much the fitness is there, fitness in terms of reproductive fitness, and also how much the present gene pool is available to cope up with the adverse effect due to climate change. We also assess the natural variation of some desirable traits related to growth and wood quality of teak, and we develop some molecular marker so that we can do the marker assisted selection, and also the clonal technique was also established. So these are all are the methodologies. We apply dominant and codominant markers AFLP and ISSR for the genetic characterization of Indian teak population from 12 states, and we collected 39 samples from 39 localities. We also perform ecological niche modeling so that we can check the suitability of the niche in the future or projected climatic scenario. The characterization of the phenotypic trait, we choose growth trait like height, girth, and breast height, wood quality trait, wood density, lignin content. And for this experiment, we didn't do the sampling from the natural forest, but we prefer garden test, and we sample the teak from the National Teak Germplasm Bank, Chandrapur, where they are having assemblage of 12 states, teak of the 12 states of India. So for the development of genomic tool, we also developed some express sequence tag based SSR markers, and that were applied for the association mapping to find out some marker which is related to the wood density and wood quality parameters. And also we did the experiment to clone the superior genotype that we could identify through our work. So these are the results. When we are considering only the entire meta population of teak as a admixed population without considering the geographical barrier, we found that the panmictic heterozygosity ranges from 0.2 to 0.3, and FST is also 0.2 to 0.22. So enough adequate genetic diversity is still there, just like a wild population, though teak has been domesticated and being planted every year by the forest department. Still a lot of genetic diversity is there, and majority of genetic diversity exhibits within the population, and we cannot say there is a clear defined variation between the cluster. This is a result of population selected from the peninsular and central India, and we are finding that some populations are having admixing of both kind of populations. Okay, so when we are considering the geographical barrier also, this kind of geographical barriers we are considering means we applied the isolation by distance. We find that the all South Indian populations are clustered together. Even some Central Indian populations are clustered together, but in a mid for high admixing there is another population. So if we see it on map of India, we find that majority of these populations are clustering together, and these one are all together, and in a mid, this is the third kind of cluster, and there is a very high gene flow among them. And direction of gene flow if you will observe, we find that it follows the direction of the monsoon wind that first during the first week of June, the time when uh, actually this is a season, flowering season of the teak as well. So we discuss is that like there is a influence of monsoon current on the direction of the gene flow. So Indian teak is a highly admixed and with very high genetic diversity being maintained by the entire gene pool. So how it is going to be changed in the future? We applied ecological niche models also with marker environmental association mapping. Teak is known as a frost susceptible species in its silvicultural properties, and the similar result we found when we applied marker environmental association mapping, that minimum temperature could be a driving factor or regulating factor for the distribution of the teak population. And also we saw this trend that after decreasing the temperature to the 15 degrees Celsius, it is a limiting factor for the distribution of the teak population in India. And also we, through ecological niche modeling, we predicted the suitability of the ecological niche. Red portions are showing the highly suitable area. Most of the area that is high suitable and going to be high suitable in the coming year 2050 to 2070, these are there in the central India, and least change we could observe to the present scenario. So it means the species. Yeah. I'm just the three, four slides are remaining. Yes. So in coming year, the projected climate change and other adverse effect is not going to affect the teak populations in India. We also studied the genetic variation among the phenotypic trait. We found that average wood density was 796 up to 800 kilogram per meter cube with coefficient of variation of 10.57%, which is very high. And we can see that at least high selection intensity is required to do the selection based on the wood quality trait. We also recorded the data of pest resistance against the Eutectona macellaris, which is known as the scrutinizer, and we found that this is a relative resistance, and we found one tree as with a high relative resistance among the other clones of the teak. So even through the association mapping, we could find one marker which was very similar to the gene from the enzyme kefil coenzyme methyl transferase, often from eucalyptus playlist, very similar and responsible and covering 4.3% of the phenotypic variation in the wood density in the teak. So and also we have identified some trees with a higher wood density and resistant also. So how we can provide it? We found that the if we are removing the seed core with the kernel, maximum rooting can be observed, or germination can be observed, and suitable. And similarly, when we are trying to do the cloning through the branch cuttings, so with the effect of rice bran extract, maximum root induction can be observed in month of May, and moisture content up to 45 to 53%. So this is a kind of part of work was done by Mandal and Ansari, and I included this as an extension of the work. So the basic achievement that we could have is first, peninsular Indian population is the center of genetic origin of teak that we could analyze, and Indian teak is adaptively fit to survive on the threat of climate change. 14 trees we could identify which are having the wood density more than 900 kilogram per meter cube, and one tree relative resistance is higher against the pest scrutinizer. So similarly, we identify some genomic marker also, which are able or highly specific to the teak as well. So after validation of this marker can be used for the marker assisted selection at the early age. And second, this thing can be verified. Yeah. So this was actually the last slide. After validation, the similar. Yeah.
Your message is well taken. Actually, you have covered a lot of work actually. And it is equivalent to 3 to 4 papers you have presented in one. I can see remote sensing, I can see genetic diversity, I can see future planning. It's wonderful. In fact, teak is something some people, I have myself a lot of questions on teak because I am fascinated with teak plantations, but because running short of time, I will contact you myself, and those who would like to interact with you, I will urge all of you to individually call and ask questions to Dr. Vivek Vaishnav. You are from Manipur right now, okay?
Yes, sir. Yes, sir.
Yeah, thank you very much for your very wonderful talk. We really enjoyed it because we have another speaker already connected, and he also is running stress on time. Dr. H.S. Vinwal, I will take liberty to invite Dr. Vinwal. Okay. Yeah, Dr. Vinwal is Dean Academics and Head City Improvement Division FRI in the University Dehradun. I invite Professor S.S. to introduce Dr. Vinwal.
Good evening, Dr. Vinwal sir.
Yeah, good evening, Dr. Sir. How are you?
Okay, I will take 1 minute, sir, and then we will listen you. Dr. Vinwal, S.S. Vinwal, who is the leading authority in the forest genetic resources mapping and its detailed molecular analysis. He has been working in the Thalai Dehradun since last 30 years. He is associated with the Uttarakhand Forest Department, Punjab, Haryana, and the Government of India. He has done the genetic analysis of the trees who are very useful in agroforestry lines, odor values, timber values, plantation programs. He has done his postdoctoral fellowship from university in the USA. He is a very practical man. He is presently the dean of Forest Research Institute in University, the academic is. I visited many times his lab, and I can say very right way that presently in forest genetic resources analysis, he is the leading person we can say in the Southeast Asia. He has visited many countries. He is a very practical man. He presently sees the all academics, all the RSC programs of the FRI University, and also he is seeing the improvement program in the country. So welcome, Dr. Vinwal sir, and it will be our pleasure to listen you. So please.
Thank you.
Thank you.
Thank you, Dr. Sir. So just give me a minute so that I can load my presentation. So my presentation is visible.
Yes, sir, visible.
All right. So thank you very much for the kind introduction. And I really appreciate to the organizers and Professor Singh for inviting me to this platform. So as a task has been given to me to speak something on the forest genetic resources, which is something new and something unique. So I will give some aspects and prospects of the forest genetic resources and the initiative which has been taken on its conservation and the management. I belong to the Forest Research Institute as I have been introduced. The first question comes, what actually are the forest genetic resources? You must have heard about the genetic resources of fishes, plants, microbes, and others. But likewise, we have got a term which we call at forest genetic resources. So as per the Food and Agriculture Organization's 2014, it is basically the genetic material which is maintained within and among the trees and other woody plant species which are of actual potential or economic value. So the scope of FGR is limited to the woody perennials. So when we talk about the forest genetic resources, we are talking about the woody perennials. So we are limiting ourselves to trees, shrubs, lianas, and the other woody perennials. So this is something the genetic diversity of the forestry species which can be of actual or has some potential value for the humanity. So what is the importance? See, FGRs are the foundation for the food, nutritional and environmental security of any country. Today, a lot of negotiations takes place at the national and the international level, particularly on the adaptations, on the ecosystems, landscape production systems. So to address these challenges, the FGR diversity is needed so that the forests can survive, they can adapt, and they can evolve under the changing environmental conditions. And at global level, after the CBD, the FGR assessment and the conservation has become a very emerging concept worldwide. If we look at the diversity of the FGRs in our country, as per the Botanical Survey of India, we have got about 18,236 higher plant species in India, and out of which around 18,159 are angiosperms and the 77 are the gymnosperms. And more than 80% of this higher plant diversity, they reside in your forest habitats. So roughly we are talking about 14,500 species. And about half of this forest plant diversity, they constitutes the FGR. So roughly it is around 7,000 species. And remaining being the herbaceous flora, that includes the soft climbers, twins, herbs, and the grasses. So if we see the terminology of the FGRs, we are confining ourselves to these 7,000 species, which are under the great threat from the anthropogenic, from the climate change, from the various other ecological issues. So as per the Botanical Survey of India, there are certain species which has become threatened. Almost like 1,700 species, they have come under the category of the threatened category. So there is a need to protect those species. There is a need to conserve those species. Otherwise, one day it will happen that we may lose some valuable species from our system. Some valuable alleles will be lost forever, which we will not be able to recover. Now, there is a great threat of extinction. Almost it was estimated that about 20% of the FGRs are facing different categories of threat. So there is a need for an effective and urgent action to halt this loss of biodiversity. Now, when we talk about the components of the FGR assessment, FGR management, first and foremost is that we need to know what the nature has given to us. So it is basically the assessments. Whatever we have, we need to survey it, we need to explore it, we need to document it. Then we need to evaluate it for the various purposes. Then we need to characterize it for some specific requirements. Then some conservation methods can be adopted. So these are some of the basic components of the FGR management. Now, the entire activity of this FGR research supports the Forest Biodiversity Conservation Program. We all know that CBD is into action. There are HE agreements, HE action plans, which was agreed in Japan, and it was adopted in the 10th meeting of the COP at Nagoya that by 2050 the biodiversity is valued, conserved, restored, and wisely used, maintaining the ecosystem services, sustaining a healthy planet, and delivering benefits essential for all people. So since more than 80% of our diversity is believed to be held in forestry landscape, so there is a need to have the reliable inventory of the status of FGRs in our country. Now, we all know that the Convention of Biodiversity, we have got 3 goals to that, that basically encompasses the conservation of biodiversity and its sustainable use. And based on these major goals, so we have got national biodiversity targets. Particularly these 4 targets, they are targeting. Slides are not moving?
No. This is first time we are seeing.
Oh, just a minute.
Yes. Conservation of biological diversity. This is first slide. Yes.
So is this moving now?
Forest genetic resources.
Yeah.
This is your first slide.
So this is now, this was my first slide.
Yes.
This is the second slide.
Yes. Third slide. Which one third?
This is the second one.
Okay. Go
So
ahead.
this was basically my second slide where I was telling about what is basically FGR, and my my statement was that we are limiting ourselves to the trees, shrubs, lianas, and other woody perennials. So they are important because they are foundation for the food, nutrition, and environmental security. They are important because they provide adaptation to the changing environmental conditions. So FGR diversity is needed so that our forests can adapt, they can survive, and they can change they can evolve under the changing environmental conditions. At the international levels, it has become very, very important these days that the FGRs are assessed, then they are conserved. So this was a slide which I was mentioning that we have got almost like 7,000 species of FGRs to which we have to do justice, to which we need to document, and to which we need to adopt some conservation methods. There is a threat status on the FGRs. Am I audible?
Yes.
There is a threat perception on the FGRs, as I said, that in as per the report of the BSI, about 1,700 species of angiosperms, they are under the category of threatened. And most 7 species of gymnosperms, they have become threatened. And there are a number of species which are endemic. So there is a threat of extinction of those species if we don't take precautions, if we don't take proper actions for the halt of the loss of this biodiversity. There are these 5 major components of the FGR management that includes the assessment, evaluation, characterization, and the conservation. So this entire activity supports the forest and biodiversity conservation programs. We all know that the vision of the strategic plan of biodiversity, which was adopted in the 10th meeting of COP at Nagoya, and it states that by 2050, we need to conserve, store, and widely use the biodiversity systems of our country. And we also know that since more than 80% of the biological diversity is believed to be held in the forest landscapes, so it is the first and foremost action that is required to have the reliable inventory and the status of FGRs, which is basically one of the major subset of the forest biodiversity. Now, the Convention of Biodiversity says that there are 3 goals, and based on that, we have got national biodiversity targets. And the genetic resources, which first time we hear sometime in CBD also, the genetic resources has been categorized into animals, trees. These are the resources which can be utilized for the food and agriculture. And our country has got a unique and oldest system of the genetic resource management under the ICR system, and for which we have got 5 dedicated bureaus. One is for the plants and the PGR, one is for the insects, one is for the animals, one is for the fish, one is for the microbes. But for the forest origin, we do not have any designated national agency that is involved in conservation, management, and development of the FGRs. So as I showed you in my previous slide like that, there are these 5 important components of the genetic resources: animals, trees, fish, plants, and microbes. For the trees, certainly there is a deficiency to have a dedicated institution which could really do work on the forest genetic resources. So based on that requirement, we have been pushing at the high authorities or the ministry level to have a National Bureau of Forest Genetic Resources in the ICFR system, but so far we have not succeeded to that extent. But at the global level, a global strategy and action plan on FGR has been prepared in 2013, and accordingly, the countries are expected to act on the action and strategies which has been which has been adopted, which has been given, which has been directed for the countries to be adopted. For the Asia Pacific region also, this action plan has been prepared, and for Europe also, this action plan has been prepared. There are 4 priority areas under the FCR action plan. First is improving the availability and access to the information on FGRs. Second is in situ and ex situ conservation of FGRs. Third is sustainable use, development, and management of FGRs. And fourth is policies, institutional, and capacity building. So there are 27 strategic priorities, out of which falls under 4 major priority areas. Now, keeping all these things into view, ICFRE initiated the FGR research program in our country since 2002, when an NBFGR cell was constituted in the ICFRE system. And since then, till 2015, it has gone to various phases, various stages. And today, in 2015, at least, we have initiated a project-based program on the FGR conservation, development, and improvement at the national level. So initially, we started it with a pilot project that was implemented in the state of the Uttarakhand. And in the second part, we started it at the national level through two dedicated National Center of Excellences, one at FRI Dehradun, and second is at IFGTB Coimbatore. Now, because the National Working Plan Code is there in 2014, and that also says conservation of genetic resources of the forest origin. So with these, ICFRE has developed the Forest Genetic Resource Conservation and Development Program at the national level. It has gone into 3 different steps. First is the pilot project, which was from 2016 to 2022. Then the phase 1 of the project program that is starting from 2020 to 2024, and the phase 2 will be starting from 2023 to 2028. Now, I'll be just giving you salient activities which were undertaken in the pilot project, which was executed in one state. So the major activities of this project were for exploration, documentation, characterization, and conservation of indigenous forest genetic resources of Uttarakhand. It was started since 2016 and almost at the verge of its completion, and wherein 250 important FGRs were studied. That includes 65 RETs, 141 trees, 29 shrubs, and 15 lianas. So this falls under the strategic priority number 1 of the global action plan on the FGRs, that is improving the availability of and access to the information on forest genetic resources. So the first and foremost thing was to upgrade our repositories, because the DD herbarium, which was established in 1906, is the oldest, one of the oldest herbarium existing in our country. So it was upgraded and it was digitized, so that this repository is made accessible to the general public. Whoever wants to use it, they can use it. Then the documentation of FGR diversity was carried out in all the 44 divisions of the state. First was to identify the range of distribution of those species, and then on the basis of the field surveys and the ground verifications, their species richness and the regeneration status of the populations was worked out. Then this way, the entire list of the rare, endangered, and threatened species occurring in the state was documented. Their inventory was prepared along with the GPS locations. Then, because this is very, very important, when we are starting working with a species, we need to have a fairly good idea about its distribution. And unfortunately, as on date, we do not have a proper species distribution maps of the forestry species. So this was the task given us to make eco-distribution maps of the 50 FGRs. And here, with the help of the GIS and the remote sensing techniques, we were able to, develop the species distribution maps with more than 70% accuracy of the important FGRs. Like here, this is the map of Myrica esculenta, very, very important species of the state. Second is the rhododendron arboreum, like that. The second strategic priority is in situ and ex situ conservation of forest genetic resources. So, because seed storage is the most convenient and widely used method of the plants, because in this we dry the seeds to low moisture content and then store it at sub-zero temperatures. For that purpose, we need to collect seeds from the wild populations, because the diverse population what the nature has given to us, that needs to be collected, that needs to be characterized. Then their germination percent, etc. All these things are recorded. Then the seeds are dried so that the moisture content is reduced to a certain level. Then the viability of the seeds are tested at the periodical intervals, and to understand how the viability, whether the viability is retaining or it's losing. Then the seed microflora associated with those wild, seeds, what kind of flora is associated with that. And ultimately, the dried samples are stored at the National Gene Bank, which is maintained by National Bureau of Plant Genetic Resources. And so far, we have been able to deposit about 100 forestry species with different accessions in the gene bank of the NBPGR for their long-term conservation. Now, because seed storage is the cheap, efficient, and very effective method, but this cannot be applied into the recalcitrant species. We cannot store the recalcitrant species this way. So for the recalcitrant species, we have to have different methods. Here we adopted the in vitro storage of the species of high conservation concern. So those species which were rare, endangered, and threatened, so they were stored in slow growth cultures or in the form of pollens. So the slow growth cultures of various species were developed, and simultaneously the pollen storage cultures protocols for various species were also devised. Now this way the ex situ conservation was adopted. Now for the in situ conservation, we need to maintain and protect the diversity on the sites. So this is the most appropriate strategy for the wild species, particularly the forest trees. Now, because these days the CBD post 2020 is under preparation, and the first draft of the post 2020 strategy of the CBD, which was released by the CBD in January 2020, and that indicates that there are 5 goals now. One is for the ecosystem, maintaining ecosystem diversity. Second is for species diversity. Third is for genetic diversity. So the inclusion of genetic diversity as a primary goal reflects the increasing recognition of the role of genetic biodiversity for ecological and economic resilience. That is very, very important. Now, as we all know that genetic diversity is important, that constitutes the species diversity and ultimately the ecosystem diversity. So we started working on the species which are economically important, which has got a wide range of distribution. Here, in this case, we took 5 important species of the Uttarakhand region. Then the sampling was carried out from the states, and the range of distribution has been shown over here. This way, this is based on the prediction modeling also. Then the hotspots of the diversity, where we need to focus our conservation priorities, need to be identified, and which were identified in this case. So this is one species, Quercus semicarpifolia. So these are the regions where we need to focus our conservation priorities. So in other way, we can say that these are the nuclear zones of diversity where we can confine our activities so that those populations can be conserved and protected for the sustenance. Likewise, we carried out this entire exercise for the other 5 species also. During the surveys, we need to catalog the various kinds of microflora, the diseases occurring in various species, what kind of symptoms, what kind of culture conditions, and the causal organisms which are associated with those diseases. They need to be characterized and then conserved in the National Fungal Culture Repository that is maintained by the FRI. Now, for the ex situ conservation, you need to establish the field gene banks, particularly the species where there is a problem of regeneration, where there is a high threat perception on those species, so that those species, their survival can be ensured by establishing them in some other location where their diversity can be captured and maintained. Now, with the success which we achieve in the pilot project, the activities were expanded across the country through the FGR first phase, which we started in 2020 and which will be going up to 2024 or 2025. It was funded by the Ministry of Environment, Forest and Climate Change. Now, in this case, now we are working with 1,000 indigenously prioritized species, and the major activities again on exploration, documentation, characterization, and conservation of this valuable genetic resource of our country. Now, the implementation of this phase is being done across all 9 ICFRI institutes through 2 centers of excellences. One is the Center of Excellence at the FRI Dehradun, which is coordinating this program in the North India. Along with, we are taking into board the RFRI Jorat, IFP Ranchi, Arid Forest Institute Jodhpur, and Himalayan Forest Institute Shimla, and the jurisdiction states where these institutes do work. Similarly, for the southern or central Indian regions, the TFRI Jabalpur, Institute of Wood Science and Technology Bangalore, Institute of Forest Biodiversity Hyderabad. So they are taking care of the rest of the part of the country and under the lead of the IGTB Coimbatore. So this is basically the program which is going on as following the strategic plan of action which was approved by the FAO. And luckily, we got some funding from the ministry to execute this program. And we are getting full cooperation from the state forest departments along with the other institutions. And we hope to collaborate with the other agencies also in doing the exploration, documentation, and the conservation programs. Because ICFRI has got the networks of the institute across the country. So that is one strength. These are the 2 nodal centers at IFGTB Coimbatore and FRI Dehradun. And these are our other institutes who are involved in this entire program. So I think that's all I have to say. This was my purpose to just give an idea what kind of activities we are taking up in the forest genetic resource management and its conservation.
Thank you, Dr. Kiran. That is a very nice way to talk. And I will open this lecture for any query, any question from the participant, especially from the VR student. Dr. Kiran, I have some not queries, but some you can say quest for the rhododendron. When I was in Garhwal University, I used to be very fascinated by the flowering season of the rhododendron. Problem with this tree is that most of the flower get plucked for making juices, rhododendron juices in the area. And the seeding will naturally properly not take place, and naturally there is a problem is there. I am talking about those species who are not more than 7,000, 8,000 feet, but those who are between 5,000 to 7,000 feet high. So what is the situation now in the Garhwal Himalaya or this Western Himalaya, the rhododendron regeneration status? Can you say something about this?
So your voice was not clear. I could not hear your question fully. Are you talking about the regeneration?
No. I am again repeating. Rhododendron has a problem that when it comes in flowering, most of the local people collect the flowers before getting into seeding. So every year they collect the flower and no seed formation takes place in the forest. And it happens continuously in the practice. So what is the situation now? Is there a naturalization problem or what is the situation?
Yes, sir, you are right. I think I got this question. In rhododendrons, the major problem you already have reflected, because flowers are collected for various purposes. The villagers and the local people, they just collect the flowers. But the regeneration in rhododendron is basically through the root suckers. If you visit to the forest areas, most of the regeneration which is coming up from the root suckers. And that's why one of the reasons which we observed in rhododendron was the diversity level in rhododendrons was coming down, because natural regeneration is affected by the asynchronized flowering, you can say, and the anthropogenic pressure. Because of that, the flowers are picked up, collected, and they are sent to the industries for various other purposes. That is one of the reasons why the regeneration is being hampered. But yes, the the root sucker regeneration is more prominent, which we have observed in rhodos.
Okay, very right. The similar questions are just review of Himal. Himal also has the same problem. The coefficient, they cut, they load the, the branches before coming to the flowering. So seeding is again the problem in that way, because I have seen these two trees are having this major problem. What you can do, what we can suggest, what we can think about these two. Himal is also having that problem of natural regeneration.
Yeah, but in Bhimal, the good thing is that local villagers, they tend to keep this plant or this tree in their farmlands, in their bunds, in their boundaries. They keep on lopping this plant, and the new VP shoots, they use it for their cattle, for their animals. In forest or in villages, you don't really observe large regeneration coming up in Bhimal. Maybe again, see, during our 5 years experience in the Uttarakhand region, because we collected 100 seeds of 100 species and their various populations, that was our target. And because of various reasons, it may be anthropogenic, it may be climatic, it may be ecological also, it was very difficult for us to get the seeds from the populations, from the trees. Either the seeds were not fertile, either the seeds were not setting up, either there was asynchronous flowering, because of which the seeds were not available for collection. So this problem is everywhere in the majority of the species occurring in the Uttarakhand Himalayas.
Thank you very much. Thank you very much, Dr. Nimwan sir. We really fascinated to listen you, and we hope that in the coming future, we will also have some collaborative, this conservation, big circle conservation work in this area, especially in Chhattisgarh, where we are having large varieties, large biodiversity in different levels. So thanks again for giving us time and giving us a very valuable talk.
Thank you very much, Doctor.
One professor has some queries.
Good evening, sir. My name is Vishwa Singh, Department of Rural Technology. Sir, I know which satellite image you used for your study purpose.
Which?
Satellite image. You used for
satellite image.
Yes.
Okay, so actually we have got our GIS experts, because a big team. We have got a GIS cell, and specifically, I'm not aware what kind of image they have used it, but they procured it from the Forest Service of India.
For species identification. Indian satellite images are not suitable for species identification, because you show so many your slides related with the species identification.
No. Species identification we have not done through the satellite data. So species identification is to be done by the taxonomist after visiting the forest areas. So Satellite data was used for making the species distribution maps.
Okay, you can write, Dr. Dilwanta. You might have some confusion. Your data was on the natural basis identification and satellite gives their, temporal and spatial distribution. Okay, thanks again, Dr. Dilwant, and we hope that in the coming future we will have again interaction with you.
Okay, sir. Thank you very much.
Thank you, thank you.
Yogesh Dubey sir not joined as yet.
कोई बात आगे फिर किस तरह से
Sessions में PPT next.
हाँ कौन सी बात
Sir, एक-
क्योंकि 5 बजे फिर एक इन्वाइटेड टॉक है
Yes. Yes, sir. Hello. Is Mr. Hitat Bhatt, Dr. Hitat Bhatt present? Dr. Sharda Dubey?
Present.
Sir, can you make your presentation?
Yes.
Okay, sir. You can make your presentation.
Okay, thank you.
Dr. Dubey?
Yes. ठीक है, मैं
Good afternoon to everyone. Respected Honorable Vice Chancellor Sir, Registrar Sir, our Chief Guest Sir, Convener Sir, Co-Convener, and organizing members, and all participants. I am Dr. Sharda Dubey from Department of Forestry, GGB Bilaspur, Chhattisgarh. My presentation topic is morphological characteristics
डॉक्टर
of
दुबे
capsule.
स्क्रीन को बहुत से स्क्रीन करो अपनी स्क्रीन को फुल स्क्रीन पे जाओ प्रेजेंटेशन
जी सर.
ये स्क्रीन आपकी फुल करो इसको फुल
वो मोबाइल से सर ज्यादा नहीं
Sir, screen is full, sir.
ओके go ahead.
Sardar sir, can you please turn on auto rotate on your mobile?
Yeah. Turn on auto rotate and keep it in landscape mode.
So my presentation topic is morphological characteristics of capsule, seed, and seedling growth performance of various species of Equilaria molescensis. So introduction part of the species. India has unique geographical features and diverse climate, which are so the different variability of the species. The genetic variability depends upon the edaphic and climatic factors. An endangered species of Equilaria molescensis, commonly known as agarwood or sasi. It belongs to the Thymelaceae family. In India, this species found only northeast India. Seeds having recalcitrant and vivipary nature. The aim of the investigation was to measure the variability of seeds, seedlings, characteristics, and germination behavior of agarwood. To identify suitable seed source of attribute characters, which is helpful for the development and tree improvement programs of suitable stand of the Equilaria malacensis. Objective of the study is selection of suitable seed source of Equilaria malacensis, capsule and seed morphology of different seed sources, seed germination as affected by seed source, and pre-sowing treatments. And fourth objective is the nature and pattern of variation in seed figure and seedling characteristics in the nursery and field level study. Growth performance and biomass of seedling is as influenced by seed sources. This one is this slide is present morphological data of the Bilaspur, Chhattisgarh. This one is slides show the latitude and longitude and altitude of the various seed sources where I was collect seeds. The maps are showing which are selected, which are collect the seeds for the which are collect the seed sources, and this one is slide is also show the morphological characteristics of capsule and seeds of various seed sources where I was collect seed like Basista, Ganek Pokhari, Hatipara, Marweng, Hybergaon, Islam, Omnagar. That is the name of the where I was collected seeds. Here I show moisture content and variability and viability of the seeds. Next slide is also so genetic estimation of the capsule morphology like GCV, PCV, heritability, and genetic advance and genetic gain. This one is so genotypic and phenotypic correlation between capsule and seed characteristics of Equilaria malacensis. This slide is also so germination percentage of the various seed sources of respective pre-sowing treatments. Pre-sowing treatment I was given thiourea 1%, 3%, and 5%, 10%, and KNO3 potassium nitrate 1%, 3%, 5%, 10% water and control. And this one is also germination value of different seed sources of Equilaria malacensis. This slide is also so RG50 rate of germination of 50% seeds are germination. This one is shown. This slide is so survivability percentage and survival percent of the Equilaria malacensis.
Dr. Dubey,
Yes, sir.
make your conclusion and some important findings if you have done.
Yes, sir. Conclusion of the study identify three best seed sources based on seedling characteristics, which is Raja Bhakshava, Sipahi Zala, Ganak Pukri were sampled. Seed source screening provides a great opportunity to the tree breeders to screen and capture natural variation for success of afforestation, conservation, and improvement programs. Suggestion of the farmers: constraint of the seeds having a recalcitrant and vivipary nature. Seeds are found only in rainy seasons. Uh, uh, this slide is show the where I various seed sources when I was collect seeds. pods of the Equilaria malasinensis and seeds of the Equilaria malasinensis. Some seeds having one and some seeds are like two or three, two seeds. In the tree, I was sampled.
Dr. Dubey, time is over, I think. So I will make it open. Any question from the audience? From Dr. Dubey?
Yes, sir.
No, thank you, Dr. Dubey. Thank you very much for your nice presentation. Thanks.
Okay, sir. Thank you, sir.
Now we have another invited talk, and the person is the director. For DNA sensing and diagnostic. Hyderabad is a very important and you can say renowned person. He was earlier in CCMP. He has worked with Dr. Rajesh, who might have heard about NET. Was the pioneer of taking fingerprint identification. You might have heard about the Manisharna white case. There is a control case. It was very tough to identify the body, and he was the person who identified by taking the DNA from the charred body and said, yes, this is my problem. So that was the first authentic legal DNA evidence used in the government documents of the court. So he has also done good work, and this he's also a person that given time us, so I think he will join within 2 or 3 minutes. When he will join, then I will request Dr. Bhaskar to say something about it. What was he has already done? So has he joined?
Yes, sir. Yamini Verma. Yes. Yes, sir.
Good evening, sir.
Good evening, Bhaskar. So we'll wait for 5 minutes?
Yes, sir. Then we'll start, sir.
Yeah. Start at 5 o'clock. Just now I finished my talk, another talk.
Thank you very much, sir, for joining us. Good evening. I feel extremely honored for introducing one of our guest speakers of this National Symposium on Biodiversity. Dr. Tangras is the Director, Center for DNA Fingerprinting and Diagnostics, Hyderabad. Prior to this assignment, Dr. Tangras was working as a Chief Scientist and Group Leader at Center for Cellular and Molecular Biology, Hyderabad. Tangras's main research areas are origin of modern humans, cardiovascular diseases, mitochondrial diseases, and male infertility. Sir has published more than 300 research papers. I would like to mention few papers of Dr. Tangras. 2005 reconstruction of origin of Andaman Islanders in science, and 2009 beginning the nature genetics on MYBPC gene cardiomyopathies. In 2009 ending reconstruction of Indian population, and this was like this. more than 4 science paper, more nature papers and nature genetics paper has been published by sir. All these papers changed the perception of Indian history. These papers attracted more than 40,000 citations. Thangaraj sir was elected fellow of Indian Academy of Sciences, Indian National Academy, and Thangaraj sir is the recipient of J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, and so on. Thangaraj sir is the board of editor of Mitochondrial Journal and associate editor of BMC Medical Genetics, BMC Genetics, Tropical Medicine, and International Health. He was also serving as a member of many editorial boards of many journals. Thangaraj sir was the president of Indian Society of Human Genetics during 2011 to 2015. Sir was the founder of the Society of Mitochondrial Research and Medicine. So with this brief introduction, I welcome Dr. Thangaraj sir to deliver the lecture. Sir, thank you very much, sir. Thank you.
Thank you, Dr. Bhaskar, for inviting me to give a talk. What I'm going to speak is about our own origin and how our practice of endogamy is affecting our health and disease. So good evening to all of you, and I hope that it's a gentle talk. It's not too much of technical. I am aware that they're all very diverse audience. I hope that you like my talk. Is this slide is visible in a full mode?
Yes, sir.
Presentation mode?
No, sir, not in full mode. Not in full mode.
Okay, this is what is the problem I'm facing. Anyway, I will not try further, and I will keep it in the regular mode.
So you can present entire screen.
Entire screen. I'm unable to see that slide. No?
No, so you can open the slide on the system, and then the three dots are there. You can just select the
Window I am sharing, but it's not showing. If I share the entire screen, then the screen would be visible again. I don't know. Let's see.
So now you open the
Slider.
slide. Yes, sir. You can follow the full.
Is it visible?
Yes, sir.
Full screen is visible?
Yes, sir. It's visible.
Okay, good.
You can move slide to check.
Sorry?
Slide transition also.
So, is it moving?
Yeah, it's moving. Great. Thank you, sir.
Okay. Yeah. Yeah. Good evening, all of you again. This topic, probably you may be wondering what is the connection between our origin and our practice of endogamy. Endogamy is the marrying within the group, and how these are impacting our health and disease. But if you see the statement of one of the well-known evolutionary biologists, Dibjansky, where he has written an article in a journal called the American Biology Teacher, where he wrote, and the title of the article is "Nothing in Biology Makes Sense Except in the Light of Evolution." So this is very strong statement, and you would realize that how important the statement is as my talk goes along. So coming to origin of species, you all know as a biologist that Darwin's work, he wrote a book called The Origin of Species in the year 1858, where he has shown about evolution of most of the organisms, ranging from orchid to whales. But what was missing in that was that how human might have originated. Okay. And there is another well-known evolutionary biologist, Thomas Henry Huxley. He wrote a book called The Evidence as to Man's Place in Nature. where he was arguing that we all descended from apes. I think I request all of you to mute your mic, and there's some background noises coming. 8 years later, after Huxley wrote a book, Darwin wrote another book called The Descent of Man, where he declared that chimpanzee and gorilla all are our closest living relatives based on the anatomical similarities which is existing between human and chimpanzee. If you look at the divergence period of both human and chimpanzee, so that happened about 6 to 7 million years. We diverged from a common ancestor called hominids, where you can see the chimpanzee and humans have diverged. And what happened during the last 6 to 7 million years? There are several species have evolved, and most of them have vanished. Okay, so the many species. And there are 3 distinct features. One is the ability to walk with 2 legs, called bipedalism. Other one is making tools, because not many animals can make tools. So we have gained knowledge to make the tools. The third one is increase in the brain size. If you look at this early species, Chandensis, the brain size was as similar as chimpanzee. But in case of Australopithecus, brain size increased to 35% of the modern human. In Homo habilis, brain size increased to 50%. In Homo ergaster, 60%. Then gradually it increased even beyond the brain size of human. In case of Homo neanderthals, the brain size was predicted at slightly larger than the human. The question is whether Neanderthals were intelligent than human or not, but nobody knows. But why this species has vanished? There are several hypotheses. One of the hypotheses given by Paul Mullers from Cambridge. He wrote an article where he has mentioned that inherent violent behavior of modern human probably responsible for vanishing of this species, Neanderthals. Nobody knows whether it's true or not, but I believe that it could be one of the reasons. But this species, Neanderthal, has given gaining more attention in the recent past. There are several studies comparing the sequence, the DNA sequence of both human and chimpanzee. And also recently, it has been shown that the 50 kb region which we inherited from Neanderthal is making South Asians more susceptible for COVID. But of course, we have analyzed, and in our case in India, it's not so. So that means there are several controversies. But if you look at what we have shown earlier, along with many others, we have sequenced 300 whole genome from all over India, all over the world, where you are seeing the dots. These are the places the samples have been collected. So the Neanderthal genome was up to 3% in most of the countries except in Africa. Okay, so all the out of Africa humans carry some proportion of the Neanderthal genome. But what is the reason for the evolution? Or how, if you take our own feature, if you compare chimpanzee and human, there are several features different. So how we have evolved and look like the way we are looking today. So as a geneticist, we believe that there's a lot more DNA rearrangements have taken place during the course of evolution. If you look at the genomics, human and chimpanzee share about 99.6% of DNA similarity, but not necessarily exactly the same as what the human has. But the genes are existing, our genome is existing, but they are existing in different places. If you take Y chromosome alone, if you see the gene called SRY, that's a pseudo-autosomal region here, and this is a sex determining region on the Y chromosome. This is present on the P arm or short arm of the human, whereas the same thing is present in the long arm of chimpanzee, again in the short arm of gorilla. Okay, you can see that. That means within this non-human primate itself, there is a lot of number 1, for example. And if you wanted to do where the human genome from one chromosome number 1 is present, but you might find that in many places, if you do in situ hybridization of a human chromosome number 1 with specific fluorescent dye and hybridize onto chimpanzee or any other non-human primate, you might see that chromosome number 1 is present in 5 or 6 chromosomes or many chromosomes. So that color you will see after hybridization in many places. So that means genome overall it's 99.6%, but same, but it is reorganized, rearranged. Okay. And similarly, you can see again the CDY, chromodomain Y, deleted azoospermia are in the long arm, in the short arm, right? And it is distributed both in long and short arm. So they all tells that a lot more rearrangements have taken place. So even if somebody don't believe such thing had happened, but by looking at this picture, where you see there are 3 brothers who born to a normal parent and having similar or typical chimpanzee-like feature. Okay, that means whatever the change has taken place, which brought us to the way we look like today, and there's a back mutation, which probably brought back to this kind of physical feature. So not only that, you can also see, and we have lost the hair during the course of evolution, but you can see the child as body full of air. Okay, this is another feature which we have gained, a loss of air, but now again there's a back mutation which brought the air throughout the body. So these are some of the things which actually proves that there was evolution. There's a DNA change. As a geneticist, we believe that both genetics and evolution goes parallelly. Whatever the genetic change that brought the evolution. Then the question is, how did the unique feature of human originate? How was the capacity for language, reason, and the culture encoded in the genes have evolved during the course of evolution? So these are some of the questions. And to address this, there are few studies which demonstrated that when compared to skull of chimpanzee and human, and there was a mutation in myosin heavy chain gene. That mutation brought more space into human skull, therefore that allowed the brain size increasing in the human compared to chimpanzee. There are a few more genes here. And there's another gene called protonorphin that is involved in learning, experience of pain, perception, behavior, and memory. These are some of the things which human has, which other species are not having. Okay, so these things helped when there's a mutation in protonorphin gene, which brought all these features. And for speech and language, FOXP2 has helped. Again, these genes are existing in human and the chimpanzee, but there are a lot of mutations in human, so that's what brought all these features. So not only that, so the way we behave, that also helped in having such kind of phenotype now. For example, if any animal, either cattle or dogs, that is in wild, we bring them from wild and we domesticate, so therefore they become very milder. But nobody's brought hairs from wild to in the place where we live, because we ourselves have brought all these features. So that kind of self-domestication happened, which made a human as a very cooperative species. Okay, now let's come to how the modern human must have arose. And it's very well established that the modern human originated in Africa about 160,000 years back. But the question is, from Africa, how did they migrate to the rest of the world? Okay, from Africa. There are two hypotheses. One suggests that the modern human migrated towards north, taking land route. So these are all hypotheses suggested by historian, archaeologists, anthropologists, linguists, geneticists, and many more specialists. But others didn't believe on that because the climatic condition in Europe probably might not have favored that migration. Therefore, they suggested that the modern human might not have gone towards north, but they must have come towards south, taking southern coastal route. So what is the evidence to say that? So the large number of archaeological evidence suggests that people lived in Australia about 50,000 years back, in Europe 39,000 years back, in Middle East 47,000 years back, and Europe 40,000 years back. But unfortunately, we don't have any biological evidence or archaeological evidence suggests that when did the modern human arrive in Indian subcontinent. There are very few studies, but it's not actually giving any evidence that when did the modern human arrive in Indian subcontinent. One such study is that the archaeologists have excavated a place called Jwalapuram in Andhra Pradesh. That was a little old study where they have shown that there's tools, stone tools, which were used probably by the modern human for killing animal or that helped them in getting the required food. But they didn't find any biological material, either bone or air, to prove that modern human actually lived in that place. One successful genetic study which we did was from the Harappan site. You know that this is a place where the modern human civilization took place. And there was a archaeologist, large number of archaeologists who have excavated the place and identified one intact bone. Okay, so we sequenced that and found that that genome has two major components. One is the Andamanese component. I'll tell you about Andamanese in the coming slides. Other one is the Iranian hunter-gatherers. The age of that Iranian hunter-gatherer is about more than 10,000 BCE. So that's very old sample. But what is interesting is we didn't get any Iranian farmer. Why it is important is that you know that the agriculture and domestication of animals started in a place called Fertile Crescent in Iran. So if this genome was found here, for example, let's take the color, blue color, then we would conclude that probably they are the one who brought the agriculture here. But since we don't find this, they might have come much later, but India itself, there would have been agriculture which was initiated. Okay, so that's the observation, but we could not get much of information because the sample is not very old. The sample is about 3000 BCE only. Okay, so again, it's not giving any clue about when did the modern human come in India. There was another study. It's one of the largest ancient DNA study. The samples have been analyzed from South and Central Asia, particularly in South Asia. We did not have much of the bone sample. Mostly it's been Central Asia, about 500 human bone samples. And what this study suggests is that around 1000 to 2000 BCE, there were Yamnaya migration. Okay, very small proportion what you see here. That is present only in the north but not in the south. And the same people also migrated towards Europe. Okay. So they are the one who brought Indo-European language here. Same way here, the European language has been has gone into Europe. So again, it's not very old sample, only 1000 to 2000 BC. So all suggests that if you look at any of the ancient sample, we are not going to get any clue about origin of modern human. These are a couple of studies near Madurai in Tamil Nadu, Kiredi and Achanallu. This work has not begun yet. However, even if you do it, we are not going to get much information because the samples are not very old, around 3000 BC. So in the absence of the archaeological evidence, so what we thought is studying the existing contemporary human population genome would reveal the information about when did the modern human migrated out of Africa. And when you consider the amount of diversity exists in India, it's amazing. And it's not exaggeration to say that India is an ethnic museum because we have collection of large number of population groups. And some of you may not aware that in India we have 4,635 anthropologically well-defined population groups. That includes 532 tribes, 72 primitive tribes. Half of them are still following hunters and gatherers lifestyle. That means they just go and hunt for food. They don't do any job, including agriculture. So that's a proportion or constitution of Indian population. So we thought that studying their genome, because they preserve their genetic material very, very in a way that they are not manning outside, they are manning within the group. So whatever the change takes place, that's going to remain in that particular population. Therefore, studying each population would give much more information. We have been studying this for last more than 2.5 decades, and to our surprise, we came to know that the first out of Africa human is in Indian subcontinent. So who are they? They are none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware that there's a typical population who resemble the features with Africans, and they are in Indian subcontinent. To give you a little more idea about that, the Andaman and Nicobar Islands, where these tribal populations are there are about 572 small islands, which is about 2,000 kilometers from Chennai and 2,000 kilometers from Calcutta. There are 6 tribal groups. Four of them typically look like Africans, like Jarwas, Great Andamanis, Sentinelese, and Wonge. There are 2 populations, Nicobaris and Champagne. They are in the southmost islands. They typically look like Southeast Asian. Okay. So we have been studying this for a very long time. This is the one which we collected about more than 20 years back. So a lot of information are in this, so I'm not going into detail of that. We collected and we started looking at different genome. In a cell, for students to know that we have 2 different kind of genome. One is the nuclear genome, which comes from the chromosomes, which is inherited from both the parents. We have 46 chromosomes. We inherit 23 chromosomes from the father and 23 chromosomes from the mother. Whereas we have large number of mitochondria in a cell, and each mitochondria will have a large number of mitochondrial DNA. It's very small, tiny circular DNA. It's only 16.5 kb in size. And the number of mitochondria and number of mitochondrial DNA is not fixed in every cell. So vary from cell type to cell type. Wherever there is an energy requirement is more, you will find more number of mitochondria and more number of mitochondrial DNA. That's why it's called as powerhouse. So For example, our brain, egg, heart muscle, where it keeps pumping the blood, will find more number of mitochondria. But most importantly, why this genome and when we can use it? We all inherit the mitochondrial DNA only from the mother. There's no paternal contribution of mitochondrial DNA. Therefore, it's useful for tracing the maternal lineage. Okay. So we sequence the complete mitochondrial DNA of these tribal groups, and based on the mutation exists in the mitochondrial DNA, one can construct the haplogroup tree. So if you compare two mitochondrial DNA sequence, this is one sequence, this is another sequence. Most of the place it's identical, but you see that there's a dot. The dot shows that there's a A in one of the sequence, and the other sequence there's a G. Similarly, there's one A here, whereas G here. There's a G here, and there's a A here. So these are the changes. So you find a lot more changes. But we take the highly conserved, evolutionarily conserved mutations and try to construct the haplogroups as what you see here. This is a parsimonious tree where this is the root of the mitochondrial haplogroup tree, and roots are L1, L2, and L3, which is present only in Africa. That itself says that the population, human population, originated from Africa. So in addition to the set of mutation, there is different set of mutation. The individual can be placed in haplogroup M. Instead of this, there are different set of mutation, they will fall into haplogroup N. Again, if you take haplogroup M alone, there's hundreds of sub-haplogroups. Similarly, there are several sub-haplogroups in each haplogroups. But if you look at the map where these haplogroups are present, L1, L2, L3 are restricted to only in India. In Europe, different set combination of or proportion of haplogroups. In Middle East, different proportion. India, you see that majority of them are M haplogroup. And you can see Australia, different haplogroup, PNG Island, different haplogroup. Then you can also see North and South America have different kind of haplogroups. So that itself says that when the populations are diverged and they are living in different isolation, there's no admixture, then whatever the mutation existing originate in one population, it remains there, it may not spread in the other population. So that's what making every population very unique. So this is very useful for tracing the population history. So what we did, we sequenced the complete mitochondrial DNA and tried to assign the haplogroup for the Andaman tribes. And these are the mutation, what you see 200, that means when you have 16,500 base pair and you start from number 1, at 200 position, there is a mutation. Similarly, 1,524, there's another mutation. 2,045, another mutation. So these are the unique set of mutation which is not present anywhere in the world. Okay. If it is there in any other world, then we can relate to them. Even this is not there in the Africans also, because the split was very, very long. That's why there's no common mutation. There are some common, but this mutation is unique to Andaman tribes. Based on the mutation, one can also calculate the age of the population. So when we calculate the age of the population based on the mutation, it comes to 65,000, around 65,000 years. So that gives a clue, and based on that we propose that the Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route, and they have reached Andaman and Nicobar Islands about 65,000 years back. Of course, this is a very old study, where we have shown that the Andamanis are the first modern human, and this is the very first genetic study showing that the Andamanis are the first modern human who migrated out of Africa. Of course, the subsequent studies showed that they further migrated to Australia, and we can still see some kind of affinities between Australian Aboriginals, Andamanis, and some South Indian groups. After this, we also now wanted to look at the nuclear genome. because after human genome was sequenced, the variation across different ethnic groups have been identified. There are about 1 million single nucleotide polymorphic markers have been identified, and they are available to use. Every single individual can be studied in one experiment, about 1 million single nucleotide polymorphic markers. So we did that in large number of human population within India. We also carefully look at the background. There are two backgrounds one can say. One is the linguistic background. So there are 4 major linguistic groups. One is Dravidians, Indo-European, Austroasiatic, Tibeto-Burman. These are the 4 linguistic groups. We also have the Andaman tribes. So based on this, we further gone into the social structure. Every linguistic groups, there are several social structures like so-called upper caste, middle caste, lower caste, tribes and primitive tribes. So we incorporated all of them so that we have a representative sample from all over India. Then we also compared that with the Human Genome Diversity Panel and HapMap data set. So what we done was, so the entire information actually helped us in reconstructing the Indian population history. So how did we do? So we did with several statistical analysis. One of the analysis called principal component analysis, where you see these are all Indian group in the plot. These are all Indian populations. This is European HapMap sample. This is a African HapMap sample, and this is Chinese HapMap sample. Now you see that by looking at these 3 groups, there are many samples, but they are in a single cluster, suggesting that genetically they are more homogeneous compared to Indians. Okay, Indians, you see that every single population is unique, right? And every single population is unique, but still they have genetic affinities with other populations. That's why there's a gradient of cluster. Otherwise, there would have clusters somewhere here and there. It suggests that India is not inhabited by single large population, whereas it is inhabited by large number of small populations. Further, what we have concluded is that these two are deep rooted. Okay, Andaman is migrated from Africa. Okay, it's very long time back. But while Andamanis were migrating, some groups stayed in southern part of India, whom we call as ancestral South Indians. Okay. There was a second way of migration. People gone again from Africa towards north, and somewhere probably in the Middle East, one group has gone to Europe, and there's a split, and another group has come to northern part of India, whom we call as ancestral North Indians. Okay, so there are several evidence suggesting that these two have genetic affinities, like skin color mutation, lactate persistent gene mutation, and R1, Y chromosome R1 mutation. They're all common between European, Middle Eastern, and people living in northern part of India. So all says that they have good genetic affinities. But what was further interesting was that we found that there was a kind of admixture between ANI and ASI. After admixing, they gave rise to many populations. So in this paper, we are unable to estimate when this admixture must have took place. To understand that, we went on to do a population from south and large number of samples from north, based on the recombination. For example, this father is from different background, mother is from different background. When they marry, the combination is always 50-50% goes to the next generation. Similarly, based on the recombination and the transmission of the genome, we predicted that initially the ASI and ANI, ancestral South Indian and ancestral North Indians, gave rise to many population groups, okay, as you see here, initially. But what happened once the population number increased, during the last 2,000 to 4,000 years, these two populations have admixed. Okay, the color indicates that the genome of both ASI and ANI have admixed. That's one aspect. The second aspect is after admixing, every single population started maintaining the endogamy, means marrying within the group. That's called as endogamy. So that happened probably for the last 2000 years. This was much before the caste system came into existence. But the caste system probably must have put more pressure on every population to maintain the endogamy. Now, what we have seen is there's a population who migrated out of Africa for the very first time in India. And in India, there were two founding populations, ASI and ANI. They gave rise to many population groups. They have admixed, and after admixing, they started maintaining the endogamy. This is some other background information. But now what I wanted to tell you is that, so what is the impact of the endogamy? Okay, they are marrying in the group, but how it's going to affect our health and disease. So that's what I'm going to give you now. So after admixture, the endogamy, to understand that, we now took another study where we have taken 2,800 individuals from 275 endogamous group. This time we did not restrict only within India. We also taken sample from other South Asian countries, including Sri Lanka, Pakistan, Nepal, and so on. And also carefully we looked at all the linguistic groups, including all the 4 major language group, the isolated languages like Andamanis, Nihalis, and a few more languages. Okay. Again, there was a background why we took sample from South Asia. Our earlier study was suggesting that the other South Asian population, in fact, carrying about 50% of the genome from India. That means India is the mother for rest of the South Asian countries. That's one. Second one, there are some common mutation which we found to be associated with our disease, is highly prevalent among South Asian populations. Therefore, whatever we study in India, it's also true for other South Asian countries. So that was the reason we thought that we'll go beyond India to look at the other South Asian population also. So what we found was that very interestingly, that there is a possibility of identifying population-specific mutation which may lead to population-specific recessive disease. They're all based on the statistical analysis. So what we did, we looked at the IBD score. IBD is identity by descent. So in simple term, what happened is that when the parents contributing the genome to the next generation, if both are unrelated, then when you sequence the individual or the child, for example, whose parents are unrelated, then if you compare the sequence, you might find a lot more variation between these two chromosomes. But if they are from the same population, I will expect that the sequence is identical and have same haplotype. It means if you take the mutations of the such of DNA sequence and the mutations are identical, that means they are in the same haplotype. Okay, so that is more common in India. So when compared to these two populations, Ashkenazi Jewish and Finnish population, most of the Indian populations, the IBD score is several fold higher compared to Ashkenazi Jewish. So what is the impact of that? The impact is that probably there are a large number of recessive mutations existing in most of the family, which has not been reported so far. The mutation which existing, what you have analyzed, most of the samples are from rural area. Even some disease exists there, and there's no way that they have been reported to health department. Okay, so most of the, for example, mostly, which some of the example I'm going to show is that there are children who are dying at very, very early age. If there's in a family, there are 1 or 2 children are dying, people may think that it's because of something else, because their belief is different. Right? So that means there's a mutation exists in the parents in a heterozygous condition. When they marry, because they are somewhere related, marrying within the population, and they're going to pass on to the next generation, therefore the disease. So that is very, very high among Indian population. So why I am showing example of Ashkenazi Jewish and Finnish population is that these two populations are well studied. You can see these are the disease, these are the genes have been identified, and most of them are autosomal recessive, and very few are autosomal dominant. So you must be knowing that if it's a dominant disease, or the mutation is dominant, out of 2 alleles, we have 23 chromosomes, 23 pairs, 46 chromosomes. 23 we inherit from father, 23 from the mother. And if you take chromosome 1, there are 2 chromosome 1, means 2 alleles. Okay. If one chromosome has a mutation, so one of the 2 alleles has a mutation, if it expresses the disease, that is dominant. If it is a recessive, if it is in a heterozygous condition, 1 chromosome is normal, 1 is having a mutation, it will not show any disease. But because of our marriage pattern, where we marry within the community, so leave away the consanguineous marriage, that means within the family. So this is within the community. So the mutation is going to increase in the community. When it increases this frequency, because they're marrying within the community, there's a chance that 2 individuals with heterozygous condition, if they marry, and this is a 25% of chance that they contribute the abnormal chromosome, both of them, to the next generation. Therefore, there's a disease. So that is very, very high among Indians. To tell you that how it happens, for example, here, this is a pedigree. This indicates female, circle, square indicates male. If you put line between these 2, that is marriage. And if you put line down and put either circle or square, it indicates their offspring. In this case, there are 2 daughters. Okay. There's a mutation here. It's an heterozygous condition. There's no disease. That means it's a recessive mutation. Okay. And she is passing on to the next generation. Both the daughters carry the mutation, and therefore there's no disease, because in the heterozygous condition, this chromosome doesn't have the mutation. I mean, one has come from the mother, so this has come from the father. Okay. Then she married somebody. She also married somebody from outside without that mutation. Therefore, no problem. It's going to the next generation. Again, she married somebody who doesn't have that mutation, so it's going to the next generation. Same way, it goes generation after generation. Again, here goes generation after generation. Then you imagine in a village where there are 100 families, and there may be 2 families may saying that they are unrelated, but actually, if you trace, they all must have come from a common ancestor. Okay, so that is very, very common in most of the populations. So now, after several generations, if these 2 things that they are unrelated, but they are carrying the mutation in heterozygous condition, if they marry, that there's a chance that they contribute the abnormal chromosomes, and therefore there's a disease. And this we have observed in many of our other studies. This is one example where there's a range of disease called mitochondrial disorders. That is caused either by mutation of mitochondrial DNA or mutation in nuclear genes, which are helping either mitochondrial DNA replications or maintaining the mitochondrial integrity, so on. There are many more nuclear genes that also leads to mitochondrial diseases. So in this case, POLG, that's a polymerase gamma, is a nuclear gene which is essential for synthesizing polymerase, essential for mitochondrial replication. If there's a one mutation introduced in the POLG, it will introduce multiple mitochondrial DNA deletion. For example, in this case, this is a wild type, means a normal nucleotide, C. And here it's heterozygous. One chromosome carry the C, other chromosome carry the T. This is a C2T mutation. And here it's a complete mutant. Both the copies are carrying T. That's why only T is visible here. So when we sequence the patient with many problems, encephalopathy, seizures, stroke-like episode, and also premature death. You can see they are dying at age of 13, 9 months, 18 months, and so on. So in that case, what happened? This mutation introduces the multiple mitochondrial DNA deletion. This is an intact wild type 10 kb region where you see there's a multiple mitochondrial deletion. So in this case, in a family setup, what you see in a consanguineous family, both the parents are heterozygous. 45 years, 39 years, father and mother, they are absolutely fine, although they carry the mutation in one of the chromosomes. It's a recessive mutation. No problem at all. But when they married, they passed on the abnormal chromosome, both father and the mother. Because the condition is homozygous, the child died at very early age with all these features. Similarly, this child. Okay. And here, this is a consanguineous marriage where relatives are married. This is non-consanguineous marriage where both the parents are unrelated. Again, heterozygous condition, 41 years, 38 years, but still you can see they are transmitting the abnormal chromosome. Therefore, there's a this child we could not analyze, and here you can see the mutation in homozygous condition. This child is no more. So indicating that these kind of recessive mutation, which is well spread in the population, and when there is a consanguineous marriage or endogamy marriage, and it leads to homozygous mutation, therefore there's a disease. So this is quite common in India. So with this, what is required is that something like what the Ashkenazi Jewish practice. They have an organization called DOR-Asia. It's a community genetic testing organization. So they go to school, collect the sample from the school children, they sequence their genome, put it in the database. When they grow to the level, they come to a level where they are ready to marriage, the boy identify the girl or girl identify the boy, then they see, the organization see whether they take out the sequence and see whether both of them carry the same common mutation which leads to such disease, which anyway they have mapped all the disease. If they have mutation, they advise them either not to marry, or even if you marry, you have to do prenatal diagnosis whether the mutation exists in the homozygous condition or not. That they have been practicing for the last several years. By doing that, they eliminated a couple of disease from the population itself. So that is very, very essential. So what we are doing now is we are targeting the population which showed very high IBD score, where we don't have whether the populations have any disease or not. Another one is where there is a population specific disease or regional specific disease. So we are targeting both and doing the exome sequencing or the whole genome sequencing, identifying the mutation and going for the population study and trying to help the population. Therefore, they will not have burden of such disease in the future. This is one example. There's a down south near Madurai, there are about 22 villages where about 80% of the population are color, and there's a majority of them that have this disease called junctional epidermolysis bullosa. Okay, and so this disease, the severity you can see, there's a skin lesions, oral mucositis, and most importantly, the congenital heart disease, which leads to very, very early death. All these children, unfortunately, have died. By sequencing that genome, we identified there's about 11 base pair deletion in LAMB3 gene. This is a very novel gene, and we screened more than 3.7 lakh genome sequence all over the world and found only one heterozygous in South Asian population, but not studied from India, but outside. Whereas in this population, the frequency is quite high. This is one population, one family pedigree. You can see that both the parents are heterozygous and the children are homozygous. Similarly, there are many pedigree. What you are seeing is this is the place. These are the individual whom we have analyzed, and most of them showing in heterozygous condition. So, in addition, we also gone to look at the regional specific disease like Handikudu disease. There's a population called Handikudu. There's a region called Handikudu, and most of the individuals are with short stature, with bone abnormality, leading to very severe pain, unbearable pain sometimes. Therefore, most of them are in a very bad shape. This is one. Another one is Udanam kidney disease. Most of the individuals from that region are with the kidney disease. They are undergoing dialysis. These are some of the examples where some of the population specific disease which are existing in those populations. We are sequencing all of them. Hopefully, we'll come up with the mutation which would help in screening the population and giving appropriate advice in terms of premarital and prenatal diagnostics. So with that, I think this last slide. I thank all of those who have contributed to study within CCMB, and there are many collaborators within India and outside India. Without that, would have not have been possible. I'd also like to thank the funding agencies, including CSIR, UK India Education Research Initiative, DBT, DST, and Medical Research Council at London. So with that, thank you, and we'll be happy if you have any question.
So may I ask the question now?
Yeah.
So this is Dr. Vivek Vaishnav from Manipur University. Thank you so much for this very elaborative presentation based on all genetic methodology, especially genetic structure. I'm also from a genetic and but plant genetic, forest genetics. And whenever this question come about first the origin of Indian population also and the effect of endogamy, I got wonder. Most of the examples are given with the example of disease or any kind of gene which is related to undesirable effect. The pedigree shown in this presentation is very similar to the recombinant inbred line that we developed for the genetic improvement of crop, but based on the desirable selection of the desirable character. So may you explain the effect of endogamy with example of desirable gene which is related to any superior trait, just like you have given example of disease associated gene, and how much how many generation it may take to lose the in endogamy the heterozygosity up to 1%. 2% are there.
Since you are from the plant background, it's very easy for you to do all kind of selections, but in the human, it's very difficult because we cannot do any such selection in human. Right?
Now means marriage, during marriage people do the selection of bride or bridegroom based on some character, no? Phenotypic character. That's why I'm saying.
No, that's what for every one generation, even if you take average of 30 years, right?
Yes, sir.
To see the phenotype, and some of the phenotypes are late onset, right? So
yeah, you are right. I got it.
Yeah,
Yes, sir.
you cannot wait for several years for just for one character. But again, recombination, you cannot manipulate anything. So where you do in animal or plant. So any experiment in human is impossible.
Now for disease we have the data. So do we have any
disease,
data about any
fact,
desirable character as well?
In disease, it's very simple because most of the other characteristic features are not single gene. right?
Mhm.
Most
Yeah,
of this is
polygenic.
single gene and single mutation. You see that individual with this mutation, there's a phenotype, right? It's very straightforward. So you can do prenatal diagnosis and do that. But whereas for any other characteristic feature, for example, tallness or well-built or disease resistant, so they all have multiple genes. So how to eliminate every species, because this is a natural thing. You are not directing of any kind of manipulation there. So therefore, really impossible.
Yes, sir. Yes, sir. Thank you. Thank you. Thank you so much.
Good evening, sir. I'm Akriti Damkar from the Department of Forestry. Your presentation and research was really fantastic. And I'm having a simple question that
Sorry, I could not hear you properly. Maybe you can come to the mic, which is where Bhaskar is. Bhaskar, maybe you can repeat the question.
Yes, yes. Thank you.
Sir, I'm having a simple question. Like, as we know, Homo erectus, Homo habilis, there is many Homo species which have been extinct now. And as we know, our biodiversity is also in threat. So I'm having a question like, in course of evolution, Homo sapiens will extinct or not? Homo sapiens in near future will become extinct or not?
Okay, so yeah, let me repeat the question. You are asking whether the Homo sapien will become extinct. Is that what your question?
Yes, yes, yes.
Okay, but considering the number of individual Homo sapiens, right? And unless otherwise, there is a very drastic. For example, now in the pandemic, there are some individual died, right? But others survived. Majority of them have survived even after infection. So in this process, we will it will select the individual who has better immunity. So one example. Similarly, there will be a selection process which will happen, and completely evolving as a new species that takes millions of years. Right? Losing all the human species, evolving completely new, that you have to wait for millions of years.
Okay.
And there's also theory, evolution of Y chromosome. People say that Y chromosome will vanish, but that's again the question. It might reduce its size. Maybe the repetitive sequence may be deleted, but the important genes like SRY, DAS, and many more, which is essential for reproduction, it remains there. So therefore, it's very difficult to predict anything whether Homo sapien will vanish or not.
Yeah, yeah. So due to COVID situation, I got this question and I asked. Thank you, sir.
Any other questions? If there is no questions, I would like to thank Dr. Tangraj sir for accepting our invitation and delivering the lecture. Thank you very much, sir. Whenever I attend your lecture, I just refresh your this one. And the results of your papers has changed the history of India. And earlier, whatever we are thinking that the Aryan invasions, everything is ruled out with your cell paper, which is published in 2019. There is no signs of ancestry from the steppe pastoralists or any other Iranians in our Indian population. So your results are fascinating, sir, and you have given so many homozygous mutations that are leading to the problem to many of the populations in India. And thank you very much, sir. Within a short time, we have requested you and you accepted our our invitation and delivered the lecture and the lengthy lecture. Maybe from the morning, nobody has given this much lengthy lecture. Thank you very much for you, sir.
Yeah.
Thank you.
Thank you, Bhaskar, and everybody. Bye.
Bye, sir.
We will continue with the presentations. Can I call, is Dr. Hitarth Bhatt present?
Hello.
Dr. Hitarth Bhatt.
Yes, can you hear me, sir?
Yes, sir. So we will have Dr. Hitarth Bhatt, Spatial Diversity, Novel Taxa and Phylogenetic Analysis of Bacteria from the Saline Desert of Little Run of Kutch, Gujarat, India. Sir, you have 5 minutes. Please, you can start your presentation.
Okay, just a minute. Can you all see my presentation?
Yes, sir.
Okay. Is it in slideshow mode? Is the presentation in
Yes, sir.
slideshow mode?
No, sir. No, sir. Not now.
Okay. I hope the presentation is visible in slideshow mode.
No, sir.
Okay.
Yes, sir, but it's not full screen.
I don't know what is the problem.
You can start your presentation.
Just a second. Okay. So, good evening, everybody. Myself, Dr. Hitarth Bhatt, and today I'm going to present my research work, PhD research work, Spatial Diversity, Novel Taxa and Phylogenetic Analysis of Bacteria from Saline Desert of Little Run of Kutch, Gujarat, India. This work was done under the supervision of Dr. S.P. Singh, UGC-CAS Department of Bioscience, Saurashtra University. So Indian deserts are least explored for microbial diversity. And so we particularly were interested in Little Run of Kutch, which is yet unexplored desert. And majority of studies on microbial diversity has been done on vegetated soil of desert, and stones. While very scarce information is available on microbial diversity from and to get novel natural products, we must look for organisms that is from unusual and unexplored habitat. And in cultivation dependent diversity, cultivable approach plays a major role to get novel taxa. And in that, isolation strategies plays a major role. So this is my isolation site, Little Run of Kutch, map of Little Run of Kutch, where 3 sites were selected. A site is Surajbari, B is Venasar, and C is Jogar. As you can see from the pictures, soil characteristic of all 3 sites are quite distinct from each other. Soil analysis was carried out and it was found that soil is saline and alkaline in nature. and further nutrient level was quite low in all 3 sites. 2 different isolation techniques were used, that is direct plating and enrichment technique. In direct plating, 2 different diluents were used, that is distilled water and Vinogradski's salt solution. Further, 5 different media were considered to trap maximum diversity. If we see colony forming unit status in all the 5 media, then it was observed that CFU count was more when Vinogradski's salt solution was used as a diluent as compared to distilled water. Then different phenotypic analysis was carried out with respect to media characterization, morphological diversity, then biochemical test, sugar utilization test, antibiotic sensitivity test with respect to 30 different antibiotics, pH tolerance, salt tolerance, and extracellular enzyme secretion was also checked. I will not go into the detail of phenotypic analysis. This is media characterization, morphological biochemical test, sugar utilization pattern, pH tolerance, salt tolerance, antibiotic sensitivity profile. Then extracellular enzyme secretion profile was also checked. In that, protease was found to be maximum as compared to amylase and CMCase. Looking at the detailed profile of extracellular enzyme secretion, we came to know that some isolates produce single enzyme, some isolate produce 2 enzymes, while some produce multiple enzymes. These are the images for the enzyme secretion of protease, amylase, and CMCase in all 3 sites. So altogether 120 isolates were there, but after phenotypic analysis, we screened them down to 87 isolates. Then phylogenetic analysis was carried out using 16S rDNA sequencing. After identification, multivariate statistical analysis was used, such as CCA analysis and cluster analysis. Diversity measures, such as diversity indices, were used. And at last, geographical distribution of the phylotypes recovered in this study was established under 4 different categories. That is cosmopolitan, if they are present in saline and non-saline, both sites. And if they are present in marine as well as other inland saline area, they will fall under the category of saline. If they are present in only seawater or sea sediment, they will fall under the category of marine. And if they are currently only known from LRK or Little Run of Kutch, they will fall under the category of LRK. So after 16S rDNA sequencing, this is the identification table for Surajbari, and we came to know that SB26 has shown very less sequence similarity of only 98.7%, which means that this strain is probable novel species, because 98.7% is considered as threshold value, or you can say that it is a species delineation criteria for bacteria. Then this is the overall diversity of Surajbari site: 2 phylum, 3 family, 9 different genera, and 20 different species were obtained out of 26 sequenced isolates. This is the identification table of Venasar, where again we found one novel species, that is VS10, which has shown only 96.9% similarity with its phylogenetic neighbor. So it can be either novel species or it can also be probable novel genus. So this is overall profile of Venasar: 2 domain, 3 phylum, 5 order, 6 family, 11 different genera, with 17 different species were obtained out of 30 isolates. Similarly for Jogad, and as you can see from the table, there are many novel species obtained from this particular site, and which belongs to different genera, that is Bacillus, Corynebacterium, Cocuria, and Oceanobacillus genus. And they all have shown less than 98.7% similarity. So they are probable novel species or may also belong to novel genus. This is overall profile of Jogad. Similarly, 2 domain, 4 phylum, 5 order, 7 family, 11 different genera, with 22 different species were obtained out of 37 isolates. These are the phylogenetic trees for all novel species. As you can see that all novel species have shown a separate cladding pattern, and that supports that these are the probable novel species. This is the spatial diversity profile of all 3 sites, where Bacillus and Virgibacillus dominated all 3 sites, and there were also some spatial genus that are exclusive to each site. Then dendrogram based on spatial diversity, where you can clearly see that Venasar and Jogad site clustered together while Surajbari is far apart. That means that bacterial genera distribution are similar in Venasar and Jogad site, while Surajbari is far apart. Then canonical correspondence analysis was done. In that, it was found that nitrogen positively correlated with Thalassobacillus, Pseudomonas and Corynebacterium, while carbon content positively correlated with Marinobacter. Yes, sir. Yes, sir. Just 1 minute. Shannon Diversity Index highlighted that the diversity was maximum in Jogad, followed by Surajbari and Benasar site. And at last, geographical distribution analysis indicated that majority of the genus that were obtained are of cosmopolitan distribution, means they are present in saline as well as non-saline environment. Some have shown saline distribution, marine distribution, and some are exclusively present in LRK, showing endemic nature. This is the overall summary. All isolates have shown broad pH and salt tolerance, and majority of isolates have produced enzymes. 4 phyla, 44 different species were obtained. Further CCA analysis have indicated that nitrogen, organic carbon, and pH had major effect, while sulfur, magnesium, and potassium had limited effect on the spatial diversity. Majority of phylotypes having cosmopolitan distribution. And one interesting observation was that phylotypes of marine distribution decreased with the increased distance from Gulf of Kutch, which clearly states that these phylotypes are of endemic nature. One of the novel species is published in IJSEM, that is International Journal of Systematic and Evolutionary Microbiology, and it was published as a novel genus, that is Desertibacillus haloalkaliphilis. The entire work is under communication in Frontiers in Marine Science Journal, and it is a minor revision right now. These are my publications, my book chapters published, paper presentations, workshops and training, fellowships, UGC BSR faculty, meritorious fellowship, as well as CSIR SRF direct. I thank my guide, Professor S.P. Singh, Department of Bioscience, UGC and CSR for providing me fellowship, and UGC CS Department of Bioscience for providing me infrastructural facility. Thank you. Thank you so much.
For your nice presentation, your work is very good. Any question from audience? Okay, thanks. Okay, no question from audience.
Thank you very much. Thank you.
Thanking you for your nice presentation. Okay.
Thank you so much. Thank you.
Now I would like to request Gopi Koladia, if you are present.
Gopi Koladia, are you there?
Sir, I want to present.
Ma'am, we will get in touch with you. Actually, we are completing the pending presentations. If possible, we will accommodate you tomorrow if you are available. For now, we are completing the pending participants. If Gopi Koladia is not present, Kruti Dagar. Kruti Dagar. Since both the participants are not present, Dr. Dilip Kumar Pal, sir, will you be able to present? Are you available for presentation?
Yes, I shall present.
Yes, sir.
I shall present.
Sir, 5 minutes presentation and then 2 minutes for question answer. So please.
Yes, yes.
Yes, you can start, sir. Sir, please unmute yourself and share your presentation. Sir, please unmute yourself, please. So to kindly unmute the first, yes, sir, that one. Yes, sir, you can start.
Stop presenting. Presenting.
Presenting to everyone. Yes.
Sir, can you
Just,
please?
just a minute. I'm trying. I'm trying.
Yes, sir, please.
Visible? Is it visible?
No,
Hello?
sir. No, sir, your presentation is not
Yeah,
visible.
just a minute. Network is slow here. Is it visible?
Yes, sir, it is visible. Kindly start.
Yes. Sorry. So, respected chairperson and respected convener, visible?
No, sir, it has gone, the screen has gone blank, sir.
The network problem is.
Yes, sir. Kruti Dagar, are you present?
Is it visible?
Yes, sir, please start.
So, respected chairperson and respected convener of this symposium. Actually, we know that India is the motherland of plants, and Chhattisgarh also is a rich source of medicinal plants. But proper evaluation is not yet done, though there are important medicinal plants are present here. Or like this, one plant is that Cleodonda infortunatum. It is a common plant within this Chhattisgarh state. It has many traditional uses in bronchitis, asthma, in as vermifuge, in the treatment of malaria. But still, it is not fully explored, or if it is explored to some extent, not fully explored, and it is not also scientifically basically it has evaluation not yet been done. So on that basis, this plant has been selected for the study in the Department of Pharmacy, Guru Ghansdas Vishwavidyalaya. So there are so many phytochemical constituents are present within this plant: saponin, sterol, different enzymes, lupirol, okay, and many phenolic compounds, flavonoids, steroids, terpenoids, fixols are present. And the plant relates, it is a long duration of work. So if you go for the just one by one stage wise, we can see that the collection of the plant from different parts of Chhattisgarh, particularly from Achanak Marg, and then its preservation, herbarian preparations, then botanical identification from Botanical Survey of India, then grinding and powdering of the plant materials, successive extraction from the non-aqueous to aqueous, quantitative phytochemical analysis, development of the solvent system, isolation of the compounds, particularly important compounds through chromatography, then its characterization through UV, IR, NMR, and then different types of activity, particularly in this communication, it is the antitubercular activity and antihistaminic activity. So this is the basic organization of the succinylation apparatus, where from extracts as well as compounds can be isolated. And this is the different methods for the summary of the extraction and their yield percentage. Petroleum ether, it is showing 3.97%, and ethyl acetate 5.15, ethanol 6.95, and aqueous 6.11%. Actually, this communication is totally based on the experiments only. And then phytochemical analysis for the alkaloid, carbohydrates, for sterol, phenols, flavonoids, and proteins and amino acids. Then glycosides. So it is the summary of the qualitative test. From this summary, it is found that these ethyl acetate extract contain glycoside, flavonoids, and ethanol extract contain alkaloid, glycosides, and flavonoids, and steroid also. So it is the some basic thin layer chromatography for the identification, and also to get the preliminary idea how many compounds may be present within this extract. And for this, two solvent system has been used in this case. One is the chloroform methanol, and other is the ethyl acetate chloroform and acetonitrile, and in different ratio. So if after that, it has been found that one compound, particularly we have given the name as S2, that is purely isolated compound. It has been found that RF value is 0.24, and other compounds has 0.47 and 0.9 RF value. And one compound is fully isolated. Its UV visible spectra and its IR spectroscopy and NMR spectroscopy was also performed. So this is the data related to the UV spectroscopy, and from that its lambda max is determined. This is the IR spectra from that functional groups present within these compounds are determined. This is the NMR spectra. It will give some identifications of this structure, as well as the number of hydrogen carbon present within this structure. So if we go for the structure elucidation, we can see that this compound S2 RF value 0.24, lambda max 240, 1.50 nanometer, IR peaks 3381, it is for hydroxyl group, 2921 for CH stretching group, 1658 for CO group, and 1605 aromatic group, suggesting the structural similarity with the aromatic compound. And NMR data is also given. It is also confirms the structure of the compound. So this is the related, as time is limited, I have to go to some extent fast. So this is the basic mechanisms of anti-tubercular activity of the traditional medicinal trend. So in this case, anti-tubercular activity of ethanol extract of chlorondine infiltrates were performed by the micropillate aluminar blue assay methods. And the details is given here. I am not going in details about this procedure due to the time and time limit. And it has been seen that compound, particularly that BD1, BD2, BD3 for different extract, and it is showing that ethanol extract is showing minimum inhibitory concentration. And then we are going for the antihistaminic activity. It has been performed by the haloperidol induced catalepsy methods. Okay, and in this case, animal was taken. Previous experiment was the in vitro experiment, but this is the in vivo experiment. And here mice was taken into 5 groups, each group containing 5. And first, all groups received haloperidol, 1 milligram per kg, 30 minutes before the drug, after the drug administration, and its catalepsy was calculated 30 in 60 minutes. And all the animal data was statistically analyzed by different statistically significant method. In this case, ANOVA followed by the Parfoni post, and the value P less than 0.05 was calculated as a significant. So it has been seen that 200 milligram kg PO ethanol extract of Dendron infortunatum shows the maximum antihistaminic activity. So data is giving here for number of catalepsy in case of different extract and their dose. In first cases, it was the 100 milligram per kg body weight, and second was 150, and third one 200. So it is, definitely it was the dose dependent activity. So this is some bar diagram showing the activity. And this is the discussion and conclusion already discussed with this different discussion previous. So this is the just summation that how that ethyl acetate extract is showing the maximum antitubercular activity, and ethanol extract is giving the antihistaminic activity, and different compounds present within this extract, alkaloids, steroids, saponin, flavonoids, might be responsible for showing this antihistaminic activity of ethanol extract of Chlorodium infurginatum. So further studies are required also to establish the relation between the chemical constituents present within this extract and their mechanism of action, and also the toxicity profile is remaining. That is under process in the Department of Pharmacy. That is our future plan of work. So these are the different references, and also some references including my research work also. This is published in Indian Journal of Experimental Biology, and that the OPEM also some it is some 1 to review article related to this plant also. So anyway, all of you, thank you.
Any questions?
Thank you, sir, for your nice presentation. Any question from audience? Great work.
Dr. Paul,
Sir,
you have given a very nice presentation,
thank you, sir.
and you tell me you have done many, phenolics, proteins, and carbohydrates, all many things you have accumulated. So how?
Yes, sir. Definitely, alkaloids are present within this extract, and alkaloid and that glycosides are present within this, and this might be the responsible for such activity. But future further confirmational studies required to establish the relation of the compound, because this is a plant extract. There may be some additive effect or synergistic effect of showing this activity. May not be that one compound, particularly alkaloid or saponin, or maybe responsible. That is the synergistic.
That is my question. That you have taken many of the compounds and make it clinical diagnosis. How you would say that even in the alkaloids there are different types of alkaloids. Even in phenolics there is different groups. So I would say that you have to be right with that this, especially this alkaloid, because alkaloid is almost many. So
yes, sir.
It is a crude way that you have given, and you are saying this is a clinical result.
Yes, sir.
You have tried good way, and
Yes,
I'm
sir.
happy that you have taken these plants produced natural way, and you are using that. I invite you to join my department for working all these works with some of the young faculty or some of the old faculty which you train.
Yes, sir.
And you screen some of the plants in the area who is not been worked on screen still.
Yes, sir.
Make a database of the phenolics of the medicinal plant groups, and I think there will be very scope for working on these lines.
Yes, sir. Certainly, sir.
Okay, any question to Palka? Okay, thank you very much, Dr. Palka.
Thanks all of you, sir. Thanks all of you for giving me the opportunity to present my work at the GGB level to the audience and the total scientific fraternity. Thank you very much, sir.
Thank you, sir, for your presentation. Along, we are joined our natural. Thank you, sir.
Oh.
Yeah, I think this is the last one, and thank you all my friends.
We need to announce there. This was the last.
So all those who have joined online, the announcement is that today's session is over now. Tomorrow we will meet at 9:30. 9:30 we will have a keynote address by Professor Uman Shankar. He is a professor in Department of Botany, Nehu, Shillong. And after that, the technical session will start and the paper presentation again will go on. So thank you all for joining us online. Those who have presented the paper, I pay our sincere thanks. And Those who have not presented papers but joined, we also pay our thanks and for your this support to this symposium, some workshop academically in a right way and successful. So tomorrow we will meet and we'll see in tomorrow. Thank you. Thanks again.